There is an exciting new breakthrough in how we treat cancer. Imagine we could obtain vital information about a patient's tumor from a blood sample. Scientists discovered that as tumors change and their cells die, small pieces of their DNA are released into the bloodstream. These fragments are known as circulating tumor DNA, or ctDNA. So what does this mean? Clinicians will be able to identify mutations or alterations in cancer-related genes using a simple blood test. This allows patients to receive more targeted treatments while also monitoring their response to ongoing treatment. That means less invasive testing, more precise and actionable results, and better patient outcomes. This is only the beginning. For this breakthrough to touch as many lives as possible, we will need a large-scale coordinated effort to prove it works. That is where Friends of Cancer Research is leading the charge, bringing together an unprecedented group of stakeholders to propel this tool to the forefront of cancer medicine. Together, we're understanding how ctDNA aligns with treatment outcomes and how it could revolutionize our approach to patient care and accelerate drug development. And we're just getting started.
Good morning. I guess that video said everything I was supposed to say. I'm Ellen Sigal, Chair of Friends of Cancer Research. We have a really important meeting today, and we're thrilled that you're all here, and we have a lot to get accomplished. This is about establishing evidence, new advances in using ctDNA. Vision without execution is hallucination. So we are trying very hard to get the evidence that we need for one goal and one goal only, to help patients. And really, we have a lot of work to do, and we're very appreciative for everyone who's come here, who's worked hard and continues to work hard. We're on a journey, and it's going to take a lot of work, but we are sure by everyone working together towards the goal of helping establishing the evidence that we need, that we will get there, and the end result will be incredibly important for all patients that are enduring really difficult treatments. So I always get the job of introducing people that need no introduction, but that is my job, so I'm going to do a little bit of it. To kick off the meeting, we have Patrizia Cavazzoni and Fleisher. You all know Patrizia. You all know her hard work. You know the effort, the tireless effort that she puts in every single day towards getting the evidence, towards working with patients, working with science, and making very hard decisions that are not always obvious, but are always right. And I'm deeply appreciative. Fleisher, the same thing. He is a chief medical officer of CMS. He too has very difficult decisions to make and works really hard towards making those decisions that are evidence and will help patients. Again, not always easy, sometimes complicated, but we get there, and we're very, very, very pleased to have them. And I know everyone is way more interested in them than they are in me, so I'm going to get on with this. I want to introduce, well, first of all, I want to thank everybody that's worked extremely hard on this. And particularly the Friends staff led by Jeff and the great people that work relentlessly every day towards getting you the evidence that we need to really further this important science. They had wanted me to say something like our fearless leader, Jeff, but I don't want to say that. I don't like that. But I am going to say our extraordinary leader, who takes everything seriously and makes a huge difference in his quiet way. He is really a dynamo, and we're really appreciative of everything he's done and having him here. That's better than fearless, right?
Well, thank you, everyone, and welcome. Fearless may not be the right word. I'm a little nervous having the chance to be able to talk with you two this morning, but thank you very much for for joining us and for giving us the opportunity to hear about some of the things that you're currently working on. I'm hopeful that we can pack a lot into a short few minutes here. I know that both of you have been very busy. And I'm hoping we can cover a couple topics. No surprise, some of the things that you both have been working on regularly make the news. So hopefully we'll be able to cover some of your current initiatives, generally at the agency, how you work together, and then really dive into some of the aspects of accelerated approval, and we'll go from there. But thank you very much for joining us. Obviously, the accelerated approval mechanism has been an important component of cancer therapy development over the last several decades, with over 200 different indications that have been approved to market through accelerated approval. And obviously an area that continues to get additional attention, and recently went through some reforms in Congress that I think we'll be able to touch on a little bit later. But before we get into some of the specifics, I wanted to ask a question that has come up recently about how your agencies work together, and some of the areas in which there has been collaboration either in the pre-market space or in the post-market space between FDA and CMS. Maybe I'll start with you, Dr. Cavazzoni, if you don't mind.
Sure. So, our work really occurs in parallel, and as everybody knows here, I mean, we have sort of our different standards, different statutes that sort of frame our decisions. We, at least when it comes to sort of drugs, I think that there's more interactions when it comes to devices for obvious reasons. We tend to sort of interact before approval or after approval, when it comes to generally potentially new classes of medications, sort of milestone approvals in new classes of medications and so on. But generally, we work in parallel, and we converge sort of as needed, or we speak as needed. We try not to surprise each other. Most of the time we succeed, and we really feel that it's important to have communication and a common understanding of the statutes and regulations, and rationale for our respective decisions, so that we are able to explain them to our common constituencies.
So I agree entirely, and thank you for this invitation, Ellen and friends. We do have different statutes: safe and effective, reasonable and necessary for the Medicare population. The amount of collaboration that has occurred in the 3 years I've been there during COVID has been unbelievable. I talked to Peter Marks continuously about what was coming, treatments, because the goal was that as soon as new treatments were developed, we had to have a plan for coverage. We did that with COVID, we did that with monkeypox, so lots of collaboration. I think what's really important is we about 2-3 weeks ago came out with TSEC, Transitional Coverage for Emerging Technologies. That actually says that a year before FDA approval, or it's not approval, it's in the device side, it's clearance, or that a year before we will start working together. We will be talking with industry, and the goal is to develop, if there needs to be more evidence in the Medicare population, that at the time of FDA clearance, that there will be a joint plan, or a plan that both agencies will find acceptable for post-marketing and for our statutory obligation of coverage with evidence development. And we really outlined that pathway in a series of guidance documents. We're still looking for input over the next 60 days, or now it's probably about 45, of what that should look like. But that collaboration is really important. I have to say, even with the elephant in the room, the Alzheimer's drugs, that we did talk before they were actually, we knew when the timing was, we coordinated, and as many of you may know, we have a CMS facilitated registry, but we've already had discussions because you can have multiple registries to meet our CED criteria, And the goal will be that if FDA for any post-marketing group, for any post-marketing study is approved, that the study also gets submitted to CMS. So that's really only for the providers and the people we care most about, the beneficiaries, patients, people, that it's the simplest path.
I'm in complete agreement. I think the dialogue and sort of the interactions are very important. Again, particularly for some sort of milestone developments. I mean, COVID, all the COVID programs were milestone
Yes.
development program. But we have, we apply, we approve, hundreds of new applications or supplements every year. I think that in all of this, it is very important that we remember that the two agencies, have different statutes, have different mandates, and that they, while talking to each other and collaborating, that they sort of maintain sort of independence when it comes to decision making. Because, for instance, FDA cannot take sort of pricing or access considerations or access into consideration when we make decisions about approval, whereby obviously those are very central to CMS's sort of mandate.
True, except for price. In my center, the decision for coverage is entirely independent of price, which is why it's under a career civil servant. But you're right, your colleague Jeff Shuren, almost weekly between Jeff Shuren and Tim Stenzel, there was a testing, because we're talking about testing in some ways, a testing work group for I think about 2.5 years that I had to see them on Zoom as we talked about novel tests and how we could ensure coverage, and that coordination was and continues to be quite amazing. I think tomorrow I have a 3:30 call with them.
I'd love to get your thoughts. Oftentimes these decisions are built upon the same original evidentiary base. So in the context of therapies that are leveraging novel endpoints, I'm wondering if you each could provide a little bit of thoughts on considerations that each of you have from your vantage point around the utilization of novel endpoints.
I'm happy to start. So it depends. Obviously, novel endpoints can be used in different ways, right? They can be used, for instance, to look for early signals of drug activity very early in development. They can be used to drive sort of enrichment in clinical trials. They can be looked at to drive sort of prognostic. They can be used as sort of prognostic markers for population selection and so on. Now, when it comes to sort of the using novel endpoints to sort of support regulatory decision making, we do have a framework to determine whether endpoints can be used to support approval or traditional approval or accelerated approval. And so there is a process of sort of data collection that needs to inform whether an endpoint is ready for prime time, be that as a surrogate endpoint, like potentially in the future, we don't know, depending on the data, could be ctDNA, or for as a clinical endpoint. When it comes to sort of ctDNA, I know that there's a lot of robust discussion. Today's meeting will be really important to advance those discussions. We are at a stage where we need to collect more information. And how do we collect the information? Well, we collect it in the clinical trials and during development. And so it's very important that novel endpoints start being collected in clinical trials that have that utilize accepted endpoints, so that then we can collect the data to see whether there are associations between accepted clinical endpoints, such as PFS, overall survivals when it comes to cancer and so on, and what could be potential future candidates for surrogate endpoints. And so we need to have sufficient data. We need to do meta-analysis of the data in various clinical trials. And it's very important that developers come and talk to Rick Pazdur and his team, because it's very important to have the right guideposts when these endpoints are included in clinical trials. So for instance, we would want them to be included in clinical trials and then reflect or are consistent with the clinical trials in which the endpoint, the novel endpoint, could be used potentially in the future for regulatory decision making. So there's a journey, and it's a journey that we have sort of traveled in across multiple therapeutic areas, and even more so in cancer. And I would say, Rick and I talk about this a lot, that it's also very important that there be sort of socialization of a novel endpoint, even after the sufficient data and the sufficient analysis sort of have taken place, so that there is a sort of a common understanding across the treatment and also the payer community that and sort of acceptance of that endpoint. Over to you, Lee.
Yeah, no, thank you. And as I think most of you know, most drugs are actually paid for by the MACs, the decision to pay for anything by the Medicare administrative contractors from a reasonable and necessary. There's very few, if any, in the cancer space, to my knowledge, maybe before my time, but that we have a national coverage decision. So, and in cancer, it's been the tradition that the surrogate endpoints and the FDA approval has been paid for. I think as we develop new surrogate endpoints, and that's really what we're talking about, it is what you said, Patrizia, the level of scientific evidence. And I think we have a robust collaboration, discussions, FDA, CMS, NIH, and really when do those get to where they really are so well linked, like they have been to date in the cancer field, with improving health outcomes, that it clearly meets our statutory ability to pay for reasonable and necessary, and to leave it to the MACs to make those decisions.
So a lot of work going into the pre-utilization of novel endpoints, but aside from the novel endpoints for accelerated approvals, Patrizia, I want to give you an opportunity to provide a few insights into how FDA is thinking about some of the new authorities that Congress has provided, namely around things like ensuring that the subsequent evidence following an accelerated approval is produced in a timely manner.
Yeah, so Congress has given us some additional tools that, from my perspective, it will actually be advantageous in expanding the use of accelerated approval, while at the same time making the data collection following approval faster and more reliable. And so the new authorities are really can boil down to two new tools. The first one is an expedited pathway to withdraw drugs when the confirmatory trials have not shown clinical benefit. As I'm sure you have seen, it can take several years to be able to withdraw a drug when that has happened. I mean, we've had a notable instance with the McKenna withdrawal. It's all in the public arena, so I can talk about it. And it took, 2.5 years. And it's a very cumbersome process, and it's very lengthy, and so on. And so having an expedited pathway will certainly help, and it will also go some way into instilling even more confidence in the robustness of the accelerated approval process for the naysayers out there, because we will know faster, we will be able to act faster when we see that the clinical trials, confirmatory trials have not panned out. There is another sort of important tool which is about the confirmatory trials and when they are going to be started relative to the approval. And based on this new authority, generally, we will be expecting the confirmatory trials to be underway by the time the drug is approved and enrolled by the time the drug is approved. Now, there can always be exceptions, and we have seen situations, particularly in rare diseases, where we may sort of uncover a surrogate endpoint for that particular disease, and it may be difficult to start the clinical trials, by the time of approval, or we may actually uncover or characterize that surrogate endpoint during the review process. However, I would expect those to be, very rare instances, and really the rule of thumb will be is that we will want to see clinical trials enrolled and substantively enrolled, ideally by the time the drug is approved.
Lee, with that idea of continuing evidence generation in the post-market space, you touched on this a little bit briefly, but given the pace that science and technology is evolving, are there ongoing initiatives at CMS that regarding fit-for-purpose evidence generation?
Thanks so much for that, and that really is what TSET's about. I'm still practicing, and I'm very familiar with the TAVR registry, which was wonderful, and my colleagues at my university say it's a national treasure, but it is really intense, and it's expensive and all, and we're really looking at how do we create a paradigm under TSET and working with the FDA where it says it's for this purpose. We actually have a defined time limit, so it's not forever, that once we reach those key elements, those key endpoints, we stop doing CED. So the studies, the way to analyze it, we, as part of the lecanemab study, we did a sample size calculations that we can start to look at it. If it answers the questions, CED gets stopped. So that's really very important. The other thing is that we're very excited about what we did in this registry. I would call it real world evidence, which is what we've, and the FDA, and we talk to them, and we talk to NIH all the time about what does real world evidence, and I'm old enough to have worked in SEER database 25 years ago for those, and it's more of an epi database in cancer, but we were able to ask the question of, did helical CT scans work that we published? And I know evidence has changed. So when we were thinking about this, the question became, can we actually merge claims data and therefore not collect the demographics, not collect the disease states? That's all in our claims, and have some very parsimonious, and it takes about 4.5 minutes. Took me 2, but if you had additional data you needed, it takes about 4 to 5 minutes to enter the data into the monoclonal for Alzheimer's disease registry once every 6 months. We'll merge it, and then the scientific community can use that enhanced data, or our colleagues at FDA, to really see this, what I would consider the future of real-world evidence. So we're very excited about this directionality. I was meeting with the national coordinator, how do we get these to be FHIR-based? How do we actually, for people who have EHRs, how can we actually make it seamless? These are the ways that CMS and all of HHS is thinking a lot about FHIR and the US CDI of can we use that to get to that real world future.
So this is a step towards the things that have been visioned around even that learning healthcare system as a potential case study here.
Well, It's an interesting question because the question becomes, could we use our quality measures not at a health system level but at an individual level that would be anchored on USCDI, we've said this publicly, that would be bulk fire based and pull them in, but also so they stay at the health system, but then give report cards back to the health system and say, you know, I learned when we used to talk to people, the best way to tell people how they're doing is a "you are here," and show everyone else on that chart. That motivates, in my mind, clinicians much more than almost anything else. And I think, you know, it might take us 5 years. Anish Chopra thinks it could be next week, but that's, I think, the future.
If I may add a couple of points. Obviously, depending on the drug, the population, real-world evidence collection may be more or less important, and that is complementary usually to ongoing sort of clinical trials, sort of data collection from clinical trials, and they really are. You know, clinical trial data and real-world data are sort of different sort of dimensions of that inform our understanding of the population, the disease, and what the drug does. We need to be very cautious, however, and remind ourselves that the real-world data have to be fit for purpose. And you know, the same data may be informative for certain disease states, for certain endpoints, and may not be as informative, or largely, you know, may not have been evaluated for use for certain endpoints and so on. And so sometimes we're able to use claims. Sometimes we need to have you know, primary data collection in registries to be able to ask to answer the questions about real-world use of a drug in a certain population. That really comes down to, for instance, you know, whether certain endpoints are collected as part of general clinical practice, or whether it is important those endpoints have to be collected on top of what is collected in clinical practice. And if you look at neuroscience, for instance, and neurodegenerative diseases, many of the endpoints that we're really interested in may not be collected. The scales may not be used in clinical practice. And therefore, there may be a need to actually do additional primary data collection if and when we decide to have registries. And you know, FDA does and has required registry. In fact, we have Lee, you brought in the sort of the Alzheimer's, the monoclonal antibodies, the amyloid monoclonal antibodies. We have asked for a registry as a post-market requirement for our latest approval. It is a primary collection registry because it recognizes that there are a lot of information that are not necessarily collected in clinical practice and or may not be billed for, which is really what claims are for. And so I think that we have to be, it really comes down to the question and the data, and whether the data are fit for purpose for the questions that we want to have answered. And it's really on a case-by-case basis.
And I couldn't agree with Patrizia more. I mean, that's the exciting part and the discussions, in that we created a fit for purpose for our coverage with evidence development, but we said any registry that would fit yours, please submit the study to us, because it will answer our studies too. So really this idea of, depending on the questions you need to answer, we can't answer some of the critical questions that your label asked. So that's what I hope that the future is, that combination.
I guess before we close out, I want to kind of get your thoughts thinking about back to some of the ideas that you mentioned in novel endpoint development and the large-scale collaborations that you've talked about in the post-market space, you know, the amount of planning that needs to go into that. And thinking about endpoint development and the large-scale types of collaborations that we'll be discussing throughout the day, how do you encourage stakeholders, and how can the agencies help ensure that the right data is being collected through some of these partnerships?
Yeah, I think it's really critical that you come and talk to us, especially when developers are thinking about collecting data on relatively new endpoints with maybe an eye out to understanding the performance of these endpoints against established clinical endpoints. Coming back, obviously, ctDNA being a really good example. The more there are upfront discussions with the agency, with the division, the better it is. Because again, it's not as simple as saying, well, let's start collecting these endpoints, because ultimately the performance of a new endpoint or a surrogate endpoint may vary depending on the population, depending on the tumor type, and so on. And so it's really important to have those upfront conversations. And then, as I said earlier, to systematically start collecting these endpoints, obviously not as primary endpoints in a clinical trials, but clinical trials have lots and lots of secondary endpoints, so that we can start developing the data sets that then can be analyzed collectively as meta-analysis, looking for associations both at the group level and at the patient level. And that's really what brings us, that's the journey that of the sort of evaluation of a new surrogate endpoint. I'm not talking about necessarily qualification, because here what we're talking about are sort of surrogate endpoints that reasonably predict clinical benefit, which is what we use for accelerated approval, which is different from an endpoint that is a validated endpoint that has been sort of determined, where we have evidence that supports that it can actually replace a clinical endpoint. So there's work to be done, but it's very exciting to see that once again, the cancer community is at the leading edge and is really charting the way for other areas, because let's face it, what we're seeing here, for instance, in more utilization of accelerated approval in areas that had been dormant for decades, where there was no hope and no treatments for decades, such as neurodegenerative diseases. We're beginning to sort of see those therapeutic areas beginning the journey that the cancer community began well over 20 years ago. And so you're continuing to be the pathfinders. So.
And absolutely, and we made a colorectal cancer screening decision recently, and we said if the FDA clears a particular screen and it has a sensitivity and specificity per the FDA, it's automatically covered. So we're actually using the FDA, how the FDA thinks. But I come back to, on Friday I will be practicing. I will probably be working with surgical oncologists, because I tend to do that on Fridays. And the question is always the patient, coming back to the patient. What does the patient need to make an informed decision? And patients are only so informed. They can only absorb. I mean, if you have Ellen, you can make very informed decisions. But most of the public needs the clinical community to interpret the data for them. So I would ask you, what do we need to make a good decision, to have a good discussion with a patient, to say, based on the evidence that you found safe and effective, what is this treatment right for you? And I think that, to me, is the key.
Well, I want to thank you both for being here. I want to thank your colleagues for your willingness to be part of these types of collaborations. It is a journey, but it's been through these partnerships that hopefully we've been able to advance in a rapid fashion the evidence that we'll be talking about later today. But thank you very much for joining us today.
It was a pleasure.
If you don't mind,
Thank you.
please join me in thanking Dr. Cavazzoni and Dr. Fleisher. Well, thank you. As we transition to our first session of today's meeting, a couple logistics that I wanted to start with. For each of the next two sessions, we'll have data presentations for two recent ctDNA partnerships that were briefly mentioned. The first will feature data presented by Dr. Brittany McKelvey, focused on an analysis from 8 different ctDNA test developers exploring baseline ctDNA levels across different cancer types. You may recall that this project was originally proposed about a year ago as part of a Friends initiative to lay out the evidentiary milestones or a roadmap to help coordinate data collection to inform the potential use of ctDNA as an early endpoint. The second session will start with a data presentation by Dr. Hilary Stiers and is the most recent data readout from the ctMoniTR project, which aims to answer the question, are changes in ctDNA an early indicator of long-term treatment outcome? Both of these projects, as you've heard, are unique collaborative efforts that would not have been possible without our project partners. You can see how many of our contributors have been involved I believe we have a slide listing everyone who has been part of these initiatives over the last couple of years. And I very much want to thank them for their time and expertise as we've worked together to develop these projects, aggregate the data, analyze the results, and present them here today. You'll hear from many of the organizations that are participating in the ctDNA projects during the panel discussions following the data presentations, which will dive a little bit further into each project and delineate next steps for the potential use of ctDNA as a drug development tool into the future. In between sessions, we'll break for lunch and have the opportunity to hear from Dr. Pazdur and 2 leaders from FDA spearheading 2 very important projects as an update of work being led by the Oncology Center of Excellence at FDA. If the first session participants could start joining on stage here, I think we're all set up, which means I've talked long enough to reconfigure things. We'll go ahead and get started. During the panel discussions today, we hope that these will be interactive discussions and encourage your questions and participation. For those of you that are joining us in person today, you'll find standing mics that will be in the aisleways, and we encourage you to use those as the moderators call on you, if you wouldn't mind just identifying yourself, particularly for the people that are joining online. And for those of you that are remote, please utilize the Q&A function at the bottom of your screen. We'll have people that will be monitoring those and be relaying questions as much as possible during our panel sessions. The slides for today's presentations will be available after the meeting. So, and please follow along online on Twitter by using the hashtag ctDNAFriends. And thank you again for joining us today. We look forward to the data discussions, and I'm pleased to introduce Brittany McKelvey, who's been the project lead for this study, to provide an overview of the ctDNA baseline project.
Thank you, Jeff, and thank you all for attending. We are really excited to be able to share some of the initial data on our baseline ctDNA project evaluating baseline ctDNA measurements across cancer types and stages, which is really a collaborative effort, as Jeff alluded to, across multiple assay developers. This is part of our larger ctDNA portfolio, so I'll take a few minutes just to highlight that. This portfolio really started with our ctMoniTR project, which my colleague Hilary will be sharing more about this afternoon. But this set the stage for the development of our evidentiary roadmap, which we shared last summer. This evidentiary roadmap really setting the stage for how can we use ctDNA to as an early endpoint predictive of long-term outcomes. And from this evidentiary roadmap, one of those evidentiary needs identified was a better understanding of baseline ctDNA levels and led to the baseline ctDNA research partnership. Again, the roadmap was really developed as recognizing the need to develop an aligned strategy for generating both the data and the evidence to support the use of ctDNA in patients, specifically here looking in solid tumors, as a drug development tool in oncology and for regulatory decision making. This includes the use of ctDNA as an early endpoint, which is predictive of long-term outcomes in patients. As the multi-stakeholder working group worked to develop this evidentiary roadmap, one of the evidentiary needs that came out of this roadmap was the need to have more robust data supporting baseline or pre-treatment ctDNA levels across different cancer types and stages. And this is partially due because we do have some knowledge gaps surrounding baseline ctDNA levels currently in the field. In the metastatic setting, there's variability across different assays and the availability of robust data at the baseline pretreatment levels And in the early stage setting, there's really a lack of robust data across different cancer types, across assays. And due to the variability and the methodologies and the assays used to measure ctDNA, it can make pooled or aggregate analyses challenging to be able to better understand ctDNA levels. And so we therefore established the Baseline ctDNA Project to help establish this evidence to support the use of ctDNA across different cancer types and across different stages, as well as to identify key questions that would be important for future data harmonization efforts. Our objectives for this project are listed here. Briefly, to compare baseline ctDNA level trends across cancer types in both the early stage and late stage disease setting, as well as to compare across stages within the same cancer type and across assays within the same disease setting. Through this information, to be able to then develop considerations and lessons learned for future data harmonization efforts across assays. For today, for our initial data, I'll be sharing some of the objectives that we met, highlighted in this red box. This includes data from 5 different late-stage cancer types. We have data from non-small cell lung cancer, breast, bladder, prostate, and head and neck squamous cell carcinoma. And we also have a comparison of early versus late non-small cell lung cancer. With this data, looking across assays, we can also start to understand assay comparisons and variability to, again, develop those considerations and lessons learned. To briefly share some of the assay characteristics, we have 8 assays that contributed data for this initial analysis, 5 of which were tumor-informed, meaning those mutations were identified first in the tissue and then were tracked in the plasma. And we had 3 that were tumor-naive, those that were detecting mutations de novo from plasma. All but one of the assays included used next-generation sequencing. We had 1 ddPCR. We had half of the assays that in their pipeline for this data analysis did not filter for CHIP, and we had others that used bioinformatic-only methods and 1 that used germline sequencing methodologies. We also, when looking at the different biomarker features assessed, see that all the assays included looked at SNVs. The majority of assays also looked at fusions and copy number variations, although there were varying LODs and analytical performance, as you can see on the right, with the median and range across assays. None of the assays that are included in this analysis looked at methylation or fragmentation, and I will note that all assays did meet the pre-analytical specifications that were proposed by BloodPAC. These 8 assay developers then contributed summary-level data for over 2,000 patients with early-stage non-small cell lung cancer and over 87,000 samples of late-stage cancer types, with the 5 cancer types denoted in the table on the right, broken down by cancer type. To briefly share some of the sample characteristics for this data, where in the table each column is an anonymized assay data set. You can see blue just highlighting the proportion for those data elements. You can see listed here looking at non-small cell lung cancer, the sample sizes, as well as the median age range, and the proportion for gender, which were largely similar across data sets. Also, I will note that while each developer could confidently note whether a sample was either early or late stage, we also had some developers who could delineate between the clinical 1 through 4 staging. And so you can see, for example, in early stage non-small cell lung cancer, 3 out of the 5 of those assays showed the different stage breakdown, although this varied for the proportion of samples within each stage category. As you can see, assay D, for example, in early stage had a higher proportion of samples with stage 3 disease, and assays E and I had a higher proportion of patients with earlier stage 1 or 2 disease, which again could impact cross-assay comparisons when we're looking at ctDNA levels. I will note, though, as you can see for late-stage non-small cell lung cancer with a lot of the dark in the unknown, as well as in the rest of our late-stage cancer samples. Many developers did not have clinical stage information for these late-stage cancer types, as well as across most cohorts, prior anti-cancer treatments, as well as recurrence and progression status were largely unknown. This therefore really impacts the ability to understand how representative this data is of the broader population, but also really thinking, given the unknown clinical pathological characteristics, how baseline is being defined in each of these data sets, and may potentially create biases as we compare across these different assays. Lastly, to further highlight a little bit of this variability, we also analyzed the timing between a patient's diagnosis and ctDNA sampling. As you can see in the box plots, which represent the median and interquartile ranges for timing, you can see for non-small cell lung cancer, it's relatively short amount of time between sampling, diagnosis and sampling, but you can see for some of those later stage cancers, the time is quite large. This could be due to many factors. It may be impacted by the intended use of these tests and when ctDNA is being analyzed across the patient journey. It could also potentially be due to the lack of available clinical data to the assay developers, which was noted by some of our developers. They may have access, for example, to only the initial patient diagnosis and not information on recurrence or metastatic progression, which would potentially impact that timing we see here. So to get into the data, first showing a comparison of early versus late stage non-small cell lung cancer. Here looking at the proportion of samples that had detected ctDNA. We can see that there is variability in the frequency of detection across these different data sets. However, if you do look at the assays that have data available for both early and late stage, assays A, B, and D, we do see that generally late stage samples do have a higher proportion of samples with detected ctDNA than our early stage samples. I will also note on this slide, and for the rest of the slides in the presentation, we are delineating between those assays that are tumor-informed versus tumor-naive or liquid-only, just for additional clarity when thinking through the analysis. For those samples that did have detected ctDNA, we also then looked at a composite ctDNA variant allele frequency metric, here showing the median VAFs. Again, we see variability in the VAFs across the assays. We see generally the late stage samples, again, having higher VAF ctDNA levels than early stage samples, although again, there is variability here. Now to next compare across these late stage cancer types that I noted earlier with these 3 assays that did have data across these 5 different cancer types. Again, as earlier with the non-small cell lung cancer, looking at the proportion of samples with detected ctDNA, we do see that within each assay, the baseline ctDNA is similarly detected across these late stage cancer types for the majority of samples. And then when we look at the median VAFs, we see again that they're relatively similar as well across these 5 cancer types, which really truly highlights the potential opportunity to use ctDNA in all of these cancer types. So in conclusion, for this initial data readout, we did observe some general trends. Firstly, showing that in late stage non-small cell lung cancer, there is a higher proportion of detected ctDNA as well as ctDNA levels compared to early stage samples. And across the late stage cancer types that we looked at, we saw similar detection of ctDNA. However, as I noted earlier, there was heterogeneity looking at assay characteristics, which includes the biomarker features that were assessed, the intended use of the assays, as well as analytical performance, as well as the heterogeneity in the available clinical pathological data, which could lead to challenges interpreting aggregated data. Therefore, additional data, as well as common data standards, are really needed to help make more robust comparisons and support future data harmonization efforts. So with that, I would like to thank all of our project partners that were integral in developing our common analysis plan used for this and helping to interpret the data, as well as those that contributed summary-level data to this work. I will now turn it over to our moderator, Mark Salson, who's Executive Director and Head of Technology Innovation at Personal Genome Diagnostics, part of LabCorp Oncology, to continue the conversation. Thanks, Mark.
Okay, thanks, Brittany. I will, and fortunate to introduce our panel here and kick us off for our first discussion session. So we'll start out, and I'll introduce our group here. James Chen, Senior Vice President, Medical Informatics at Tempus. Greg Jones, Head of Precision Diagnostics at NeoGenomics Laboratories. Anand Pathak, Medical Officer, US FDA and CDRH. And Eric Peters, Executive Director, Clinical Diagnostics and Data Science at Genentech. So, really before we jump into the data, and I think, going back to as we were thinking about this particular work stream and, really charting out, what are some of the next steps for clinical evidence generation, within early stage disease and even late stage disease, as we're thinking about the prospective generation of data sets, but a lot of us have existing data sets, and how can we leverage those to better understand how we move forward. So wanted to start out with just the concept for this type of work stream, where why is it important to be able to assess baseline ctDNA levels across different cancer types, stages of disease, and across different assays. So Jim, we'll start out with you here.
I think the basic concept is that these ctDNA assays need to be as fungible as possible to the greatest extent possible, so you can help the greatest number of patients to the greatest extent possible. Without this fungibility, I don't think we're doing patients a disservice. And secondly, here, part of this idea of a baseline and understanding that not just the assay characteristics, but as Brittany pointed out, the clinical pathologic characteristics that really drive some of the heterogeneity. Without these understandings from a lot of these retrospective analyses, it is very hard to look forward and design studies or design retrospective studies without these data elements kind of in tow.
Yeah, thank you. I think we all have certainly appreciated that as we were starting to map this out and look across data sets for how can we bring these data sets together in a meaningful way? And maybe segueing a little bit into that, Anand, I know that you've had some thoughts around this as well. But as we started to identify different cohorts and understanding ways in which we can bring those data sets together, what are some of the technical and biological challenges associated with that?
Well, first of all, I'd like to say that ctDNA holds a lot of promise in advancing patient care. And I really believe in that, and I believe that it might give you an early signal for disease, right? But the evidence has to be generated convincingly, and baseline in itself is important because the trackability of a patient may depend on whether or not there's a signal present at baseline, and also baseline is emerging to be known as a prognostic factor. But in terms of the technical and biological aspects of ctDNA and the assays, there are many technical issues, right? Like there could be tumor-informed assays, tumor-naive assays. There could be ddPCR assays that only target a few genes. And then there's the issue of how do you subtract the germline, right? Like, is the germline being addressed appropriately? And then there's the issue of chip. Is chip being addressed appropriately? Some people are using bioinformatic pipelines. Some people are sequencing the Buffy code. Some people are doing nothing for chip, which may give you a false signal. So those are some of the technical issues we sort of are concerned about. And then there are really important issues with the analytical sensitivity and the LOD of the assay, right? And that's all analytical issue, and it's the clinical issue, because the clinical cutoff determines many clinical parameters of how a patient might do. So those are the technical aspects. And from a biological perspective, different tumor types may have different shedding, right? Especially in early stage disease. So that's really essential to lay the groundwork and understand what that is. And there are numerous publications that show that in early stage, you don't have 100% detectability of ctDNA. But besides the tumor type, the tumor stage is very important. And besides the tumor stage, I think timing of the collection of ctDNA with respect to the intervention or whatever is going on before baseline sampling is really essential to know. So that's sort of my perspective on the technical and biological aspects.
Yeah, very, very helpful. And Greg, do you want to add on to that from a...
Yeah, well, follow on with what Anand has to say. I think as we look back 4 years ago, it was mainly comprehensive genomic profiling. And now we're in the era of molecular residual disease. And we're looking at, rather than looking for a specific marker like KRAS G12C or such, we're looking at just the ctDNA as the analyte. And often what we're doing with tumor-informed assays is aggregating the signal that we see across it. So a lot of the technical challenges are going to be on the intended use of the assay itself, the intended use population, especially as we start to move into the stage 3, stage 2, stage 1. We'll be moving away a little bit from what I'm seeing here, which is probably mainly adenocarcinomas, stage 3B and stage 4, into ones where squamous is more incorporated, where we'll actually probably pick up squamous a little bit as we get into stage 1 more than we will pick up adenocarcinomas. So I think a lot of this, the technicalities of this do play into it, but also the definition of what the assay is, what the intended use population is also going to be very important for efforts like this, because these are very, very valuable efforts. But in order to get the best information out of it, we really need to know what goes into it so that we can then pull the proper data out of it.
Yeah, great points. And Eric, you maybe have the exciting but challenging job of figuring out how we incorporate this into therapeutic development. And so based on the data Brittany presented, were these results expected? Were you surprised?
Yeah, it's a great question. One, I just really would like to congratulate the Friends team for bringing this together, because I think it is, we've seen in the literature a fair amount of promise with respect to ctDNA. What this shows us, essentially, is that at a baseline, we can detect it, particularly in the metastatic space. So I think having that fundamental, I think to Greg's point, starting from a CGP, and now we're getting quantitative with our ctDNA. So I'd say this recapitulates what we've seen in the literature, but at a broader scale, right? So we've seen the number of samples that have been tested here. So it gives me a lot of hope that, particularly in the metastatic space, we'll have a reasonable shot at, if we can detect ctDNA at baseline without a lot of, the beauty of ctDNA, but also the curse is it's a sample of convenience, right? So a lot of these were probably samples that came in for other reasons or were in a freezer, right? So seeing that we can, in the metastatic space specifically, see ctDNA and baseline across all those indications at that high level with such disparate assays, I think is very heartening. So in that respect, it was great to see this. The early stage, I think, we'll probably get more into this both later in this discussion as well as in panels. Intended use is very much going to matter, right? What are we trying to do with our ctDNA analysis in the metastatic space? Is it internal decision making? Is it potentially, an accelerated endpoint? In the early space, is it prognosis? Is it something else, right? So I think the intended use and how we utilize these assays, this is showing great promise with respect to metastatic space. Now we need to do something similar in early space. But overall, I think the results were, great to see because they recapitulated the kind of smaller individual study we've seen either from assay manufacturers or from pharmaceutical companies in a given indication with a given assay, put it together for this meta-analysis, and I think it looks very promising.
Yeah, very, very encouraging findings. And maybe kind of adding on to that and segueing a bit into some of the heterogeneity, and Greg, we were catching up on this before the session started. And it was a topic of several meetings as this work stream was coming together. But as we think about different types of assays, particularly in the early stage settings for assessments of molecular residual disease, a variety of different cell-free DNA features that could be utilized for detection and quantification of ctDNA. And so do you see there being, in early stage and late stage opportunities for harmonization for how, we measure and report ctDNA from an assay perspective?
Yeah, I think you can see from the data here that harmonization is going to be a major requirement so that we have the proper data elements that we're collecting, so that whether or not we feed it into AI eventually, or we're just doing meta-analysis and looking at things, the data elements really should be defined. And for some of the harmonization, a lot of us are here also in part of the blood pack, which is another group. We're working on harmonization of recommended data elements so that we have a guideline for publications, for setting up clinical trials, et cetera, what data you collect, how you define the assay that you have, so that this type of analysis is going to be a lot more, how do I say, parsable, where you have the elements all defined as well. And as we move forward, as we get through, as the years walk on, there will be efforts from other groups to try to harmonize maybe the output of things. There's a wonderful effort right now from FNIH for harmonization of the CGP type of assays in order to give a standard out there so that everyone can then evaluate that based on the performance of the standard. Now, that has nuance to it because you have hybrid capture assays, you have amplicon assays, and things like that. So they're taking a lot of effort to assure that this type of standard is actually very important. So I do think with the consortiums out there, being part of these consortiums and working through that, another thing in BloodPAC that we're doing is the analytical validation protocols. It's been published already for an analytical validation for CGP, and we're soon going to have an analytical validation protocol for tumor-informed MRD assays out there. So all of these efforts will help to try to bring together a framework for the consistent definition of the assay, as well as what data elements do we need to really collect in order to facilitate efforts such as what Friends of Cancer Research have done.
Yeah, and again, I think there's a number of ongoing initiatives, and I think those are helpful to highlight, particularly as we think about moving these types of analyses into prospective or interventional settings. And maybe to that end, picking up on what you had discussed, Eric, around intended use of these assays, and Jim, maybe around some of the vision for how we could start to think about implementation of ctDNA as an early endpoint, which I think there's a lot of excitement around. Do these data help to highlight specific cancer types or stages or assays where I think ultimately probably where we would all like to be is to have this as something that could be applied across tumor types. But maybe more pragmatically, is there a logical place to start based on the evidence to date and how we could maybe advance the field more quickly?
Yeah, absolutely. So from the data that Brittany presented, you can see that the top 5 are not surprising. Lung, breast. Some of these being up there, as well as in the late stage setting. That's really a offshoot of what our clinical practice is right now and where reimbursement was. Right. So that's not really surprising. I'm a sarcoma medical oncologist. I don't see sarcoma up there. So I think we have a lot of work to do outside of the highly published, and I don't want to pick on common diseases necessarily, but we do have a plethora of information about them to a certain extent. What we don't have are the smaller diseases, which really are underrepresented. So I think we have a lot of work to lay the groundwork for understanding a little bit more pan-cancer, but that's going to take data collaboration, and kudos to Friends for putting this together. And I just want to echo what my colleagues here have also said, is really just understanding what those look like. Lee had mentioned registries earlier. What do those look like? What are the data elements that are required? And I go back to, it's not just the data elements, it's also really understanding the nuances of the clinical pathologic analytes that go into it. For example, timing of collection from time of diagnosis, right? Those are really quite important. What does their metastatic burden look like? Is it enough that they're metastatic, or are there nuances there as well? So both across tumor types, but also within tumor types, there is heterogeneity that needs to be sussed out. Simply adding additional data elements is helpful, but there are efforts such as EMCODE and other ones that are trying to get to the clinical context that we participate in. There's also efforts such as Genome X, which are looking at how do you transmit this information in a robust, consistent way. So these are more in the retrospective at the setting for data sharing purposes, but I think we need to think about this a little bit more holistically. Otherwise, we're really going to miss key findings. And again, a little shout out to the rare cancers, which are often overlooked. Yes, we're making progress, but I think we can make a lot more progress as a group.
Yeah, great, well said. And maybe taking that to the next step as we think about developing what the blueprint for trial designs look like going forward, from a pre-analytical, clinical pathologic, and longitudinal and clinical outcome perspective. Eric, as you think about therapeutic development, clinical trial design, does this effort and this data set help to define how you think about setting those studies up so that 5 years down the line we have the foundation of data that we need to advance how we might think about ctDNA as an early endpoint?
It's a great question. I think it really, in a lot of ways for therapeutic development goes back to intended use, right? Is this for internal decision making? In an early phase trial in a metastatic setting, are we going to use this data, to adjudicate stable disease patients, right? Patients that we're not really sure, the scan. What I would think, and that can go all the way from that kind of internal decision making to, putting together a robust data package that we can have with discussions, with the agency, so that we could, find a way as an accelerated endpoint. So I think it really depends on that. What this baseline data really allows us is to understand, now where should we go deeper, right? And I think it is things like, obviously, clinical pathological characteristics are going to be super important here. Timing, which I think was fascinating seeing this baseline data, particularly in the breast samples, looking at the timing from diagnosis to draw, right? We need more data there, right? What does that look like? We know breast is a low-shedding disease, but we're still detecting it. So how do we utilize that even in the metastatic space where breast cancer trials take a long time? We have to really work together to make sure that we're collecting the right samples, but also going deep with respect to what does the scans look like? What are we learning from kind of all the clinical data that we can, so we're properly interpreting it disease by disease, stage by stage. So I really see it as kind of a, what stage of drug development are we in terms of early stage, late stage, and then kind of what cancer type are we looking at? And to go deep there, I think this unlocks the ability to take this data and know where to go. And in the metastatic space, absolutely, we're detecting it. So we'll see more about the module 2 this afternoon. That's a great start. I think in the early stage space, we also have to factor in clinical practice, right? So a tumor-informed assay in a disease where we're not getting the resection specimen often is going to be difficult. So we also have to think about it from the assay perspective. What makes sense in clinical trial work, right? And if you're getting a cystectomy, that's probably going to be fine in terms of having enough tumor material to do a tumor-informed assay, and then get the sensitivity that we need. So I think a lot of this just comes down to really calling where we want to be in 5 years, and then working backwards from that. Right?
Yeah, great points. And maybe that also ties together some of the discussion from the prior session around what makes sense to do from a clinical trial standpoint, from a real world evidence standpoint, and then how do we bring all of these data together in a way that really demonstrates the robustness of
exactly. And I think that's a great piece of ctDNA, is that we can learn a lot. It's because CGP exists already, we can learn a lot kind of for free, so to speak, right? Yeah. From registries, from testing labs that are doing this day in, day out, working through kind of how do we get the clinical pathological features from either the EMRs or from real world data sources, so that we can tie those two together. So if we were doing this 10 years ago and we were doing this without the advent of CGP, I'm just more excited about it because we already have those systems in place. How do we process samples? We can probably accelerate this much more than we could have 10 years ago because of CGP being so common.
Yeah, and I think that was evident as we were starting to put together some of these data sets through the working group, where we had a sense for the data elements we wanted to capture. We created these comprehensive lists of what would be great to have.
Yes.
And then as we started aggregating these data together, realized we have a lot of gaps across these studies. And then when you look at kind of the intersection of everything, we actually in aggregate have quite a ways to go. And so Anand, as you're thinking about meta-analyses that we'd like to be able to perform 5-7 years from now for prospective studies that are just getting started, what are some important factors and considerations that we should think about?
I thought about that question, and the two key themes that emerge in terms of the poolability of data for meta-analyses is one, the assay, right? Are the assays fungible? Are they interchangeable? I mean, first of all, some of these assays we saw here had LODs that went from 10 to the -4 to 10 to the -2, and we don't really even know what the clinical cutoffs were for these assays. So, so basically, there needs to be some type of harmonization of assays moving forward to make this data more poolable. One possible solution, which I would advocate for, is getting regulatory grade reference standards that you can run across labs and establish that there's consistency across labs in detection. Another issue at hand is the output of the assay, right? Some assays have an output of mutant molecules per mil, some have VAF. Can we harmonize sort of the output of the assay? So that's sort of some of the aspects with the assay required for poolability and better interpretability of the meta-analysis. And the second part is the clinical context, right? The clinical context should be similar between the different studies being used, right? So there should be a similar or standardized timing to when the ctDNA is collected. And there should be a definition for what is baseline, for example. And there should be sort of an understanding of prior treatment or the presence of recurrence, right? So all of this data needs to be carefully collected in a prospective study moving forward. And to Eric's point, yes, this data does look really good for the major tumor types for late-stage disease, but we might be interested in early-stage disease. We might be interested in other rare cancer types for late-stage disease. So if we're able to nail down the assay specifications and the clinical context, right, that would really help moving forward a prospective analysis, prospective meta-analysis.
Yeah, thank you. And I think we're running close on time here. So I think want to kind of wrap up with, based on the vision for building out the evidentiary roadmap for ctDNA as an early endpoint. Based on these data, some of the discussions we've had, what are the next steps? Where should we go from your perspective? I'll just start with you, Jim.
Yeah, so I know a lot of my colleagues have mentioned this already, but I think it could really be summarized: data standards, data harmonization, and data sharing, right? These are kind of the key elements that will be required for us to move forward as a group, and truly it's a group effort, because I don't think anybody here in the room will have a monopoly on all the data and understanding what to do with the data. One of the things I'd like to really point out is that real world data, that retrospective data, is going to be always the largest set, and we must figure out ways to use that to the greatest extent possible, again, so we can help our patients, no matter what their setting is. There are a lot of different consortia. Again, I'll plug for Codex. MCODE, for example, is already working with EHR vendors like Epic to pull in some data elements directly through the EHR. There's additional ways of doing this, so we don't necessarily have to be stuck with traditional registries. I want to just make sure that we're really cognizant that there are ways of doing direct feeds to the EHR to really supplement and because obviously we want this to be fit for purpose, but the trick is we're not always sure what the purpose is, right? The purpose changes over time. So you do need a way of sustainably looking longitudinally. You do need a way of sustainably changing your purpose, molding your purpose based on the different use cases. So I think that's going to be really the challenge. And as we think about data standards, data harmonization, that also applies to data transfer standards electronically through FHIR messaging, through other existing standards. So I know I sound like a little bit of a broken record here, but we really need to move our way of thinking about data aggregation into the future, and I think that's the only way that we're going to be able to solve a lot of these problems.
Yeah, great points.
Well, I will follow up with that is I think we need alignment on these data elements, and I think that's going to be the next step that's going to be very important. I come from the diagnostic side of things, and there are nuances with the assays. It's really important that we understand, what the LODs are, what the chip filtering and all those other things are. Plus, as we're hearing from the clinical side of things, the importance of defining the the population that you're studying and a lot of the nuances with that. So I think, what I've learned from this, some of the lessons incorporated here is really let's get some alignment on the data elements, because I'm thinking that this is, or I'm hearing that this is the most important thing that we've learned from this, this effort here.
Yeah, I agree totally that there needs to be, well thought out data elements, and there needs to be efforts to utilize, retrospective data, but also generate prospective data using established data elements, right? Not only for the assay characteristics, but also for the clinical characteristics, right? Yeah. And that gets to my two major sources of concern is the heterogeneity generated by the assay itself and the heterogeneity generated by, the different clinical studies moving forward. So if there could be, some control or harmonization around that would be very helpful. And one of the things about ctDNA is that it can have so many different uses, right? So maybe we need to, instead of looking really narrowly, maybe we should look really broadly at what are all the different potential uses and how do these assays fit in, and how can we make progress in terms of patient care utilizing this very, very promising biomarker.
Yeah, I smiled when you said, there are so many use cases to ctDNA. I think that like the sample of convenience being a blessing and a curse, I think that's also a blessing and a curse. We could think about things like in the future, subtyping disease based on ctDNA, staging, all sorts of things that I think are super exciting and can help the patient at the end of the day, right? I think going from a land of can we detect actionable biomarkers via CGP, by ctDNA to where we are now and where we'll be in 5 years is just, it's a whole different ballgame that absolutely needs coordination and collaboration, right? It's not something any of us can do alone. I think from a pharmaceutical developer perspective, we're trying, one, to get these therapies as quickly to patients as possible. And in the metastatic space, I think we'll have a great opportunity to utilize ctDNA to do that. But if we think about early stage disease, it could be, patients that don't need treatment, patients that are cured with curative intent, radiation or surgery, they shouldn't be getting treated, right? So I think as we, and even there, that's something we need a lot of coordination on and thinking through the assays, the clinical data that we'll collect both in trials and through registries and real world data sources. So I think it's really, an exciting field. It can be overwhelming because you can think of everything from early detection to metastatic CGP as ctDNA. So as we go forward, working together and defining exact intended use and how we can generate the most robust data sets to push that intended use forward.
Yeah, thank you so much. I think very, very well said, and I think, collectively, the, diagnostic, therapeutic development, medical and regulatory perspectives gives us a lot to think about, as we chart out the next step. So, want to thank the panel here, and I think we do have some time for, questions. If, anybody wants to kind of go up to the microphone, please just say your name and affiliation.
Yeah. I'm Vic from the NIH. So I have a lot of respect for people working in the advanced disease stage, but to me, it wasn't news that patients with widespread cancer all over the body have ctDNA, detectable. What I was struck by in Brittany's presentation was the early stage data suggesting variability from 2% with one assay to 90% in another assay. Now, does that mean the emperor has no clothes, or am I being overly negative about those data points?
I would like to think that there's a little bit of over-negativity there, partly because a lot of the clinical data was missing, number one, and so it was very, very hard to harmonize it. I think that's kind of the, we don't know if it's not right, just frankly speaking. All we can report is presence or absence, but again, things like timing, tumor burden at time of initial diagnosis, things like that were all not there. And as you know, practice patterns are extremely different between academic medical centers and the community. Was there a time bias from one site? None of that was stratified for. And so I think it's very hard to say whether or not that it's extremely variable or just modestly variable or not variable at all without being able to dice this a little bit further. Unfortunately, as you saw too, if we diced it even further, let's assuming we had that, we're simply not powered to do so. So again, it goes back to we simply need more data and better data to be able to finally answer that question. So I'm not negative on it. I'm very hopeful that truly these assays can be fungible and help everybody, but I think we have a lot to learn.
Charles Baylog, I'm from Northern Virginia Community College. All this sounds well and good, but what happens when you get an actual treatment out of this? Who gets the patent and who determines it?
Anyone want to take that one?
That's beyond my pay grade.
Yeah, I mean, I could take it from a pharmaceutical perspective. I think what we're looking for is, I can understand kind of the question with respect to technology development and how one, absolutely, yes. Yes, I think there is and what's been heartening to see is from our diagnostic colleagues how much the willingness to work together. I think one aspect is when we think about advancing beyond SNVs and methylation and fragmentomics, which wasn't part of this baseline effort, there's going to be, and we can throw in machine learning and things that we'll learn that have no kind of physical basis on the actual molecule of DNA, might get a little more tricky.
And just to kind of follow up on that, the rising tide kind of lifts all boats here. And I go back to that analogy. I think a lot of this, as a public good, a lot of this is being developed in the academic setting, in public-private consortia such as this. And so I think this data will be public. The standards have to be public. Yes. Data sharing has to be public. Yes. This can't be a secret. I think we're way past that. Really. Those days are long gone. You cannot work in a silo. So I think when you talk about who's going to get patent, well, if it's done by consortia, everybody will, right? Everybody will benefit from it. And so I really do think that a lot of those kind of old ways of doing things have gone the way of the dodo bird. So that is at least my hope, and that's where we see the trajectory going.
Do a follow-up. I'm just thinking how IEEE does their stuff to determine who gets what, and I was wondering if you had the same sort of procedure. The Institute of Electrical and Electronic Engineers, basically they come up with the standards for our cell phones instead of Federal Communications
I wasn't smart enough to get a double E, so I can't. I wish I understand that standard particularly, but happy to take that offline.
All right, we are monitoring our online chat for our colleagues who are joining virtually, and so someone submitted a question about the importance of chip filtering, and so recognizing in the data we had variable, how they were accounting for chip in their pipelines, the impact of different approaches, and how that could potentially impact interpretation in the data we're seeing.
Well, maybe I'll take that one. I think it's very important to filter for chip, and we don't really know at this point how the bioinformatic pipelines perform versus what would be the gold standard would be Buffy code sequencing. And if you don't filter for chip, there's a potential that you get a false positive signal, right? So it's something that we're really concerned about, and we think that these collaborative groups can help address and establish best practices for.
Yeah, the only thing I'd add, I think it's a great point, and maybe go to intended use a bit more too, is in early stage, probably, absolutely necessary, right? In late stage disease, yeah, probably depends a little in terms of what that impact would be.
Right. Also the age of the patient, right? Because,
Absolutely.
And especially as we get into monitoring, as we go from one time point to another, that's really where we're going with this, is as a surrogate endpoint, as a response to therapy, all the things you can do with ctDNA, that understanding the chip, whether or not you're following that as a monitoring, you really need to have that. So chip filtering is a very important consideration for especially the early stage cancers, where we're looking at this as a recruitment tool to be able to pull them into a ctDNA positive patient as a clinical trial, or we're looking at it monitoring. You must know the chip status.
Hi, everybody. Diana Marino-Vega from AstraZeneca here. I wanted to speak a little bit about that timing that we saw across the different cancer types. And there's a study, we put a poster together that evaluated pretreatment variability of ctDNA. So it's important to ensure that that baseline sample is as close to the start of treatment as possible to minimize any type of fluctuation that may not be controlled for. But I'm just wondering, like, with the data that we currently have, can we also talk about depth and how much, because I think that what we don't know is what we can't see. So the question is, how much do we need to see to then be able to know how deep to go? And I think that's a question that's a little bit related to the level of sensitivity that a tumor-informed approach can give you compared to a tumor-naive approach. And as Eric was saying, and I know that he's shaking his head in agreement, that has a lot to do with fine-tuning what is it that you're willing to give up if there is tissue available versus not, and that sensitivity that will come to it. And related to the previous question with CHIP, also, the tumor-informed approach, is there an extra need to filter out the PBMCs or not? So I just wanted to kind of, it's an everything question, as you could probably hear, but I'm just particular thinking about how much further in depth should we be able to go to be able to make more informed choices?
Well, I think it's very important that we really have a good understanding of the assays themselves. We're looking now at epigenetic footprints being able to help us out with early detection, perhaps in molecular residual disease as well. So having some understanding as to maybe the dynamics of what happens with epigenetic once you have therapeutic pressures on it, what happens there, what changes in there. I think just having that depth of understanding of, okay, this is an MRD assay. Okay, great, but it's an MRD based off of SNVs and indels. Okay, that's one understanding of depth. And you'll have another MRD assay maybe that's based off of epigenetic type of thing. Might have a great sensitivity with epigenetics. You get an extra increase from that, but what are the nuances with it and things? So we do, in my mind, we do need to get pretty deep in understanding that. And this is, again, getting back to the data elements, I think that's very important for us as well to have that defined so that we can really make some decisions from that, because you might see different dynamics with epigenetics versus SNV indel type of detections and going on. So yeah, it is very important.
And I'd just like to say, great question. The timing is going to be very important. So we really need to understand better what the actual right timing is to take the sample. And with respect to clinical treatment, clinical parameters, but also given the many different uses of this biomarker, right? You may actually need to fine tune your sensitivity versus your specificity depending on what you intend to do with it. So you can actually, with some of the technologies that are emerging, have really, really high specificity and sensitivity at the same time. So the field looks very promising moving forward.
And just one thing to add to that, as we think about the clinical guidelines, as CGP moves forward earlier and earlier, and to time of diagnosis, we really talk, I'm a sarcoma physician, as I mentioned, I can't really do my work without upfront CGP. right, for diagnostic purposes because of the rare tumor issue. So my patients, the timing is always very short. So if you think about as diseases, as we learn more and more, and we move this earlier, I think the timing issue will become less and less of an issue. And at least that's where I think things are heading, and that's my hope. So in the future, maybe a year or two from now, we'll be having a very different conversation.
Okay, well, I think that's all the time we have for. So again, thank you to the whole panel and for the questions. And I think we'll move on to our next session.
In the foyer outside of the ballroom. There are identical buffets on the opposite sides of the ballroom, so if you encounter what seems like a long line, don't be shy and hit the other one. Please plan on being back here in a half hour. We're going to reconvene at 12:30. You're welcome to bring lunch in here and eat, but at 12:30 we will reconvene for an update from Dr. Pazdur and the Oncology Center of Excellence. So we will see you then. Thank you very much. With what?
Overall response rate.
Oh, in the next session.
If everyone could please be seated, we'll be getting started in 1 minute.
Okay, please be seated. If your actions inspire others to dream more, learn more, and become more, you are a leader. That's John Quincy Adams. Rick Pazdur, you are a leader. You're an inspiration, and the work that you do with your extraordinary team is always for the right result, and that's the patient. And you inspire innovation, leadership, and have to make very hard decisions. And they have to be based on science and the need for improvement in care, but they have to be evidence-based. So you are, we're looking forward to a spirited conversation. I'm sure knowing you, it will be spirited. You are joined by Harpreet Singh, another leader and inspiration, and Valerie Jensen from the FDA. And Rick, it's all up to you now. Make it interesting.
Oh my. Well, Ellen, some people may love me, some people may hate me, okay? So I'll leave it at that.
That's not your job to be loved or hated.
Yes, I realize that. So we have planned we're the dessert folks here on stage. Yes. Okay, we're like the chocolate chip cookies or the souffle for dessert, so to speak. So I thought we would do kind of a hot topic type of thing because we only have a half hour. One I'd like to talk about is the new draft guidance that came out on in vitro diagnostics that Harpreet will address. And then the other, being so topical, is the issue of drug shortages, which has been a major issue in oncology. And we're joined by Val Jensen. So I'm going to ask each one of you to introduce yourself. And then we'll start, I think, first with the in vitro diagnostic issue, because I'm sure we'll have a lot more discussion on the issue of drug shortages. So Harpreet, introduce yourself, and then we'll go to Val.
Okay, well, thank you for, spending your dessert break with me. I'm Harpreet Singh. I'm a medical oncologist. I'm a division director at the FDA. I work with Rick, and he also is my boss. And I regulate the division that houses all of lung cancer, many rare tumors, and pediatric tumors as well. Valerie.
Hi, everyone. I'm Val Jensen. I'm Associate Director for Drug Shortage at FDA. So I'm in the drug section of FDA. And I've been in this role for over 20 years now. So when we started our program back in 1999. So it's been an extremely challenging role and one that we're very passionate about. So thanks for having me here today.
Yeah, I was reminiscing with Val that we began on the same year, and we've been through many, many drug shortages. Although the emphasis is now on carboplatin and cisplatin, this is not the first drug shortage or series of drug shortages that we faced throughout the years here, both in oncology and other therapeutic areas. So let's start with the first.
Yes.
That's the in vitro diagnostic.
Yes, Rick mentioned a new guidance which has been published June 20th of this year, so about 3 weeks ago, or has it been 2 weeks? And it's, our summer special. This guidance describes a pilot, a companion diagnostics pilot based in oncology. I'm really excited about it because, Rick, we have a problem, okay? We all know this, but let me just name the issue. The issue is that the current regulatory paradigm under which we operate, which is basically this one drug, one test model, correct? That in these biomarker-driven strategies, these biomarker-driven approaches, drugs which target something like FGFR, EGFR, KRAS, et cetera, it is the regulatory expectation that for each drug that is approved, that there's one diagnostic test used to identify, select patients for safe and effective use of that drug. Well, practically speaking, this is just incongruent with what's actually happening in the real world, in academic settings, in community settings, in oncology practice. What we know is actually happening is that multiple locally developed tests are being used to enroll patients on clinical trials, which are largely unregulated, unwieldy. And so at the time that the clinical data comes to us at the FDA for consideration, often we do not actually have appropriate and the amount of data, the patient samples, et cetera, to bridge, so to speak, all these local tests that were used to this one central diagnostic test. So what happens then? Well, here we're faced with a problem. And since 2014, so almost 10 years now, we've had the leverage to approve drugs without a test. And so we've done that 40 times. And what happens then? Then the problem magnifies. So then we do this when we believe that approval of the drug, making this drug available to patients, the benefits of this outweigh the risk of approving absent a one singular companion diagnostic. So we put this drug out into the world, absent a companion diagnostic. 40 times we've done this. And how the patients are selected for these approved drugs are based, again, on these unregulated, locally developed tests. And so enter the solution. One of the things I love about working with Rick and at the FDA is that we do try to address issues, problems that we see. And here you again see just an incongruence in the regulatory paradigm and what's actually happening. And patients, I think, are being potentially subjected to tests which may not actually be properly identifying them. So it's about patient safety. We think it's a public health issue. So here we have this amazing pilot, which will last for 1 year in its initial phase. It's called the Oncology Diagnostics Pilot. It's only about 9 or 10 pages, so take a gander. It's on our website, NCDRH's. And what we're asking is that we plan to accept 9 individual sponsors into this pilot. This does not limit us to 9 drug development programs. It's just 9 companies that we'd like to work with, big and small, based on the characteristics of their program. Ideally, they would come to us, you would come to us prospectively with your biomarker-driven drug and associated clinical trial and your diagnostic strategy, and we would work together to establish minimal performance characteristics that will be used with these locally developed tests. So the locally developed tests that will be used or you plan to use, you would work with these labs to make sure that they meet these minimal performance characteristics. And at the time of drug approval, all of these characteristics will be transparently publicized on OCE and CDRH's website. Therefore, with the drug's approval will come basically a set of characteristics that providers can use and labs can use to ensure that the patients they're selecting are in fact meeting these characteristics and are appropriate for use with these drugs. So trying to bring the regulatory paradigm closer to what we know is actually happening and keep patients safe.
And I think we had the initial discussion with friends at one of the meetings, and that was almost 2 years ago, right? It's at least 2 years ago, which shows you how fast government works here. I think also we really want to emphasize that this is a pilot. We want to get experience, and that's part of the issue here. And it is really looking at 9 sponsors, not 10, according to our
no,
lawyers.
We have very strict
It's 9,
PRA,
okay,
yes.
like not 10,
9.
okay. And I don't know what happens if we have 10 and not 9, but maybe somebody turns into a pumpkin or something. But
you denied acceptance.
Yeah,
so that's what
but we are asking sponsors, if you do have an interest in pursuing this, please contact us in your individual review divisions to discuss it. Obviously, this could be fitted retrospectively, but we'd like the experience to be more prospectively, because that's how we feel if this is a successful pilot, it will be rolled out into a prospective discussion with the sponsors at the time that they come in to discuss their pivotal trial, what would be the minimum performance criteria, and then this will be applied forward in the clinical trial. So there's none of this bridging that has to happen between what was used in the clinical trial versus what an approved test would be. However, I'd like to emphasize that this does not preclude somebody coming in for an approved test, a 510K submission here for a submission if somebody wants to market an approved test. And here again, we would expect that there would be an interest in some of these approved tests being done because not everybody would use one of these individual laboratory tests.
Yeah, I think in selection of the 9, we are looking for a variety of sponsors, tumor types, technology, IHC, PCR. So we're looking to really gain this experience in this year, and hopefully the pilot is successful and we, are able to move it forward.
Okay. Well, let's turn to Val, and drug shortages.
Rick, are you taking questions?
Oh, do you want to take?
Quick clarification here. 510K.
I'm sorry. Well, you could. I'm sorry. PMA. I'm sorry. Okay. Sorry. Okay. PMA. I'm sorry. Okay.
Okay.
Val,
Sure.
tell us about drug shortages.
Sure, and I know all of you are well aware of what's been going on with the cisplatin and the carboplatin, the methotrexate, all very, very critical shortages right now. So I'll just let you know kind of what happened there and what we're doing about it. You're probably aware there was a large company in India, Intas Pharmaceuticals, and they market under the name Accord in the US. So that firm actually had an inspection at the end of last year, an FDA inspection. Multiple quality problems were found. In that case, we would want to work with the company to continue production while they fix the issues. In this case, they decided to shut down, and so that was something we definitely were not, was not in our plans. We didn't expect that. We didn't want that. However, they did, and so at that point, we began working with them to resume release as well as resume production under a program. So they're actually doing additional testing, doing additional oversight to make sure that the drugs coming into the US are safe. And at the same time, we've opened our toolbox. Everything that we can possibly do, we do. Working with the other 4 companies for each of those drugs. They each have a total of 5 companies for each market. Working with the other companies to ramp up. But of course, this company, unfortunately, was a very large market share for cisplatin. So it went into shortage. It really was unavoidable. The other things that we do, we offer to the companies that are approved, we offer anything that, anything that we can expedite. So if they need a new line or new supplier, we're doing that. We also look to overseas sources. So we did find for cisplatin a company called Qilu in China, and they're marketing now under Apotex is their US distributor. So that product is now being imported from China. We had one import come in a couple weeks ago. Another one will be available this week, so we'll be posting that. So we'll have that import, and I should say when we do an import, we carefully evaluate that product. So we're not only reviewing the manufacturing facility, doing an inspection, we also do additional testing. We even did FDA testing just to make sure that that product's safe for US patients. So you'll see that product on our FDA website, the Qilu import, and as I said, we'll have more available this week for the import. And in addition, we'll have additional supply that we're working on. We went to old application holders that used to market and asked them to come back on the market. And so sometimes companies see a need there, sometimes they don't see a market. In this case, we will have some additional suppliers coming back. So that's good news as well. We do expect this supply to continue to improve over the coming weeks for all 3 of those drugs. So we're continuing all efforts on that front.
One of the things, obviously, this has not been the first drug shortage, and this has occurred primarily in generic drugs, or exclusively almost in generic drugs. What is it about the generic industry that is kind of the root cause of this issue? And I think this needs to be explained.
Absolutely, and I know Rick and I worked on shortages way back when I first started with drug shortage. So I think it is an ongoing problem, and as Rick said, it's really a problem affecting older generic drugs and especially injectables. So the reason for that really, we had a 2019 report that we published at the request of Congress, and the reasons that we found when we researched really are mainly economic. So it's an economic issue. Older drugs, they're not profitable, they get discontinued for newer products. There's a lack of incentive for companies to not only continue to make them, but also to invest in quality. So some of these manufacturing sites, like the one in India, where the quality really had gone downhill, there just isn't an incentive to maintain that quality to it needs to be. So overall, I think people are looking at solutions. We're seeing, a lot of, unfortunately, due to this horrible shortage, I think people are seeing this, as something that we need to look for solutions for. So many different solutions have been put out there. I think one is looking at maybe having stockpiles, having certain drugs that are put into kind of a rotating stockpile.
Strategic reserves.
Yes. Oh, that's a better word.
Don't use the word stockpile. Okay.
That's much better. But looking at also ways, that's right. Looking at ways to maybe incentivize these firms to either come into the market and stay on the market and not discontinue these products, which we continue to see.
Because it's very interesting, the difference between the innovator industry and the generic industry, which points to some of these problems. Whereas when we talk about pricing of drugs, obviously it's with the high prices of drugs, it has to do with the innovator drugs, and this has been a diatribe for many people to harp on the high cost of drugs. But when people take a look at the generic industry, really these drugs are commodities, basically, and it's really, to paraphrase one of my colleagues, a race to the bottom of what is the cheapest drug and price of the drug. And when you're looking at that type of pricing situation, a competitive pricing situation, this does not lend itself to quality manufacturing, in a sense. Somebody was telling me that people spend more on a McDonald's dinner for their family than for cisplatin, for example. So it's really a kind of a race to the bottom, which then leads to, obviously, poor investments or absent investments in quality manufacturing process, which does not, which makes the issues of quality manufacturing become more and more apparent, so to speak.
Yeah.
Okay. How many people work in drug shortage? Just give us an idea of what goes on your daily basis, so to speak.
Sure. So I manage a staff of 15, and that sounds like...
So we have a big commitment.
Yeah, definitely. Well, and we actually, if you look at all of the different groups working on the shortage in FDA at one given time, so we have, of course, we bring in our experts, our physicians, our clinicians, our chemists and microbiologists, whatever the issue is, we have experts on call. So at any given time, there are probably around 25, 30 people working on a given shortage. Mm-hmm. So.
May I ask, Valerie, because I get these emails often as a division director. Every time there's an anticipated drug shortage, I get an email saying it is expected that something has happened in this supply chain, and we're going to have a shortage of this drug. And it seems very preemptive, and it never makes it to this stage where we're seeing with the platinums. You talked about this one company in India. I guess, is it that fragile that, for the platinums, I guess, going back to this economic issue, that one company shutting down could cause this nationwide shortage?
And it's not only nationwide, there's international issues here too.
Correct, yeah.
I was in Europe recently, and they were facing not the severity of the drug shortage, but certainly a drug shortage.
That's true. This is international at this point. And to your point, you would think with 5 companies making a drug that you would have, you'd have 4 other companies to step up if one goes down. But it really, with the older generic market, it's really not that way, because things are made on, multiple drugs are made on the same lines, and so it's really hard to increase something because it will be at the expense of something else. So the other companies just were not able to make up that shortfall.
Yeah. Yeah, and like this, if I remember the statistics, and correct me if I'm wrong, about 80% of all drug usage is in the area of generic drugs, whereas if one takes a look at the expenditures, it's only 18%. So it's a very lopsided situation when we have that compared the generic drug industry versus the innovator drug industry, and rewards for innovation, but certainly not rewards for producing commodities that are really essential for the treatment of cancer, of patients with cancer. I'm just wondering if this occurs. We know it occurs commonly in oncology because we are dealing with injectables, and those are usually the ones that are most prone to bacterial contamination problems with quality manufacturing compared to production of tablets or pills, so to speak. But are there other therapeutic areas that you've been involved with, and what are the issues there sometimes?
For sure, and yeah, unfortunately, this is not just limited to oncology. So we've seen emergency medicine, anesthesia, antibiotics, you name it. I think anything old and injectable is prone to shortage. And that, as you mentioned, Rick, most of our shortages are injectables, and most are generic.
And it also probably reflects that even though many of these drugs are older, they're still commonly used, okay? Whereas in perhaps some other therapeutic areas, when an innovator drug comes out, some of the older drugs just fall by the wayside and are not used. But obviously, cisplatin, carboplatin are backbones of testicular cancer therapies, of ovarian cancer, lung cancer therapies, head and neck, so a wide range of diseases.
Yeah, definitely.
Any other issues that you want to talk about before we open it up to questions? Because we have about 10 minutes. Yes, Ellen?
We're all deeply concerned about this.
No.
No.
We're all really concerned about the shortages, and all of us are trying to do something about it and staff. We're an economy agency. So the question is, do you need or want additional authorities? Would that make a difference?
Well, the question that Ellen posed is, do we need or want additional therapies for these drug shortages? And the short answer is, as federal employees, we cannot comment on that because it would be looked at as our interfering with the legislative process here and proposing legislation. I think something has been done, so I'll answer that question tangentially. I think the oncology community, okay, has really asked us to step up to the plate and has asked government to step up to the plate. Valerie mentioned this concept of this strategic reserve. Obviously, we've done this in other diseases and other areas. Would it be reasonable? Yes, it would be reasonable. It could have the effect of establishing a floor of pricing rather than just going down to the absolute lowest price and ensure that we don't run into this problem. Obviously, as with every other reserve, whether it be oil, whether it be any other drug that we're talking about, stock would be rotated through so there would not be drug shortages, drug wastefulness, et cetera. So, we can't comment on this, but I think the oncology community in general has favored something like a reserve that would take place and allow us to have a maintenance of these therapies, and may be beneficial to the generic drug industry also to ensure not as what necessarily is the rock bottom price, but what is a reasonable price for these drugs, which may in turn actually encourage other manufacturers to come online and stay online, so to speak. Agreed. Other questions people might have? Oh, what a silent group. Yes, sir.
Just shaking my head at this whole thing. Couldn't we use the same model we use for our crops to make sure there's enough, that there's kind of a buyer for it?
Well, as I mentioned, these are commodities, okay, not that dissimilar. Yeah, so I think this is, one of the things that needs to be looked at. It's a very complicated issue because obviously when we're talking about the drug industry, it's not completely a free market environment because there's a separation between the buyer, the ultimate patient, and the company that produces the drugs, and that would include wholesalers and other people in the supply chain, so to speak, which adds another dimension to this. Because one would expect, if we were dealing with a very simple free market system, if the supply went down, the price would go up astronomically, and that would be reflected in a free market system, which is not exactly what we have here. Other questions people have? Hi, can you guys hear me? I'm Martha Niagu from AbbVie, and I actually don't want to ruin the discussion on drug shortages, but I did have a question about the in vitro diagnostic guidance and the proposal. How complex can the proposal be if we have a target, for instance, that is tested both by IHC and potentially PCR? Is that acceptable if a clinical trial addresses all of the above?
Yes, that would be great. We'd love to have multiple technologies. And Rena Phillip is here, and I failed to give her due credit. She's actually she formerly was with CDRH, and now we have her in OCE, and she is the point person for the pilot, and her email address is on the front page of the guidance, but she shook her head yes.
But we'd encourage you to come and speak to the appropriate review division.
Yes, of course.
With a proposal.
Yes.
That would be possible.
Yes.
Other questions people might have? That gentleman has a question.
So my question is also related to the IVD and the newly published guidelines. So I read it several times, and one question that is not clear to me is that if I have a drug that's selected by the FDA, get into this pilot program, and do we still need or is still required to develop a CodeX instead of just have?
No.
Okay.
No, I'm glad you brought this up. So if you are one of the illustrious 9 accepted into this pilot, we would ideally, again, work with you prospectively. There are some retrospective considerations that we will take. But if you're working with us prospectively for, within a clinical trial, a biomarker-driven strategy that would otherwise call for a singular companion diagnostic, by acceptance into the pilot, you would not pursue that. You would pursue ensuring that the locally developed tests meet minimal performance characteristics that we would help you establish. At the time of drug approval, you would not have a companion diagnostic. You would have listed on the website, OCE and CDRH, the minimal performance characteristics used in the plethora of locally developed labs. In turn, they would be then clinical trial assays. They were used in the trial. And that would go, and that would be how providers would in turn select patients to be treated with this drug. I think we have time for 1 or 2 more. I'm not sure about time.
There's a question from online about
let's take that. Let's be equitable to the online participants.
Pilot, maybe building off of what you were just saying, I think one question was around when thinking about the various assays that are used in testing patients in these trials, what happens to those assays where you do not have enough data to make a qualified assessment of the performance?
That's a little technical, and I think that online person, thank you for the question, but we would have to work with them. What is enough? I don't know. We expect there to be missing data, as there always is, upwards of 50% at times of missing data. So the degree to which we would accept would be highly dependent on the actual drug development program, the performance of the drug, how much data is missing. So unfortunately, I can't answer that without more specific information. But I encourage that that person asking that question to come talk to us.
Hi, Harpreet. I don't know if this is on.
Yes.
Yeah. Okay, now I can hear myself.
How do you plan to enforce minimum performance characteristics in any test? Like, can anybody just say we meet minimum performance characteristics?
Enforceable in the sense that, I mean, currently we utilize enforcement discretion, which means for locally developed tests, which means exactly what you're referring to. We use discretion to not regulate all of these locally developed tests. Frankly, it would be very, very difficult. So the question is, how would we enforce?
So effectively,
address that.
I just want to point out that you're not ensuring that these tests have any kind of quality. You're just publishing minimum performance characteristics.
Well, I'm going to take slight issue with that. I mean, the whole point of the pilot is actually to improve patient safety. We do see it as a safety issue. Certainly, we know that the regulatory paradigm and the practice of medicine, our regulatory expectations, are not universally adhered to, or even maybe 50%. But the idea is really to make patients safer by at least providing the minimal performance characteristics of the locally developed tests. That is the intent. And it's our hope that we do make patients safer.
But the issue, if you're talking about in the trial, so to speak, this would be kind of in almost an eligibility criteria that people would agree that this is the test that they would be using that would meet this minimal performance criteria for the trial. Afterwards, how do I have the assurance that somebody uses the correct dose and not uses half dose of medicine, of the dose of the medicine? How do I ensure that people don't enroll people that have liver failure? I mean, not enroll, but use the drug on somebody that has liver failure. This is what falls into the practice of medicine, so to speak. If somebody is using an inferior test and selects patients that should not be getting the drug based on a invalid test or something that does not meet the eligibility criteria, that falls within the practice of medicine and the malpractice situations that physicians always face. Okay, folks, it's been great, and we're on time here.
Thank you.
Thank you, everyone.
Okay, well, thank you, Dr. Pazdur. We certainly appreciate you and Val and Harpreet being here today. And thank you all for your continued leadership. Obviously, these are cutting-edge, current, and important topics. As we transition to the second data presentation today and move around the stage here, we'll be focusing on the most recent module of the ctMoniTR project. As you may recall, step 1 of the ctMoniTR project was a prototype to combine data across 5 initial trials as a proof of concept. The current step 2 of the ctMoniTR project is aggregating patient-level data from over 20 recently completed clinical trials. These have been bucketed into 3 research modules, the first of which we'll be discussing today includes data from 8 clinical studies of TKI-treated advanced lung cancer. I'd like to invite the session 2 panelists up to the stage, please. I think we're ready for you. And again, thanks to all of the project partners for enabling and driving this work forward. In particular, the team from Cancer Research and Biostatistics, which serves as the independent analysis center for the project, and Naveen Zarifa, who has been helping us implement the project since the beginning and will be our moderator today. But first, I am pleased to introduce Dr. Hilary Stiers, who has been the project lead for this initiative, to walk us through the results from Step 1, Module 1 analyses today.
Thanks, Jeff. Thanks, everybody, for being here. Really excited to share these new data with everybody. As Jeff alluded to, our ctMoniTR project asked the question, do changes in ctDNA reflect response to treatment? And as the project began in 2019 with ctMoniTR Step 1, which was 5 clinical trials of 200 patients with advanced non-small cell lung cancer treated with an anti-PD-L1. From this aggregate analysis, we saw that strong decreases in ctDNA from baseline to an on-treatment time point associated with improved outcomes. And so we've expanded, like Jeff mentioned, to more clinical trials, patients, and tumor types and therapies. Module 1 is our advanced non-small cell lung cancer with TKI. Module 2 will be advanced non-small cell lung cancer with either anti-PD-L1 or chemo. And finally, module 3 is kind of our catch-all with advanced solid tumors or anti-PD-L1 with anti-PD-L1 or TKIs. And so the focus of today and the data I'll be sharing comes from our module 1 analysis. We had 8 clinical trials with 8 different TKIs and 1,015 patients that were represented here. We did have 5 different assays that were used to analyze the ctDNA levels. And in addition to harmonizing across different assays, we harmonized the data across different time points. Just a little bit of a schematic, the index date for the analysis was treatment initiation or randomization. We defined baseline ctDNA as a ctDNA time point up to 14 days before index. And then our on-treatment ctDNA T1 was up to 10 weeks from the index. Across the trials, the cadence of ctDNA and first RECIST assessment varied, so this was another thing that we were harmonizing across. For a patient to be included in our aggregate analysis, they had advanced non-small cell lung cancer. They were treated with a TKI. We had a few different TKIs, EGFR, ALK, MET, and REF. They also had to have long-term outcomes. All of our patients had progression-free survival data, and the majority had overall survival data. And the analyses that I'm presenting today are for the overall survival analyses, but the results are similar for progression-free survival. As for the assays, we had 5 different assays. About half of our patients were analyzed using a ddPCR assay, and the other half were analyzed with an NGS assay. The limit of detection for NGS assays was a % VAF of 0.1 to 0.5, so again, variability there. We worked with our project partners to establish 3 research objectives. Research objective 1 looked at early changes in ctDNA and how that associated with long-term clinical outcomes, similar to what we did in step 1. Research objective 2 focused on whether changes in ctDNA complement first RECIST or BOR, best overall response. And research objective 3 asked whether if we combine changes in ctDNA with first RECIST, do we see improvements in associations with outcomes. We accomplished these research objectives as a collaborative group that included representatives from pharmaceutical companies, diagnostics companies, FDA, and academia. Cancer Research and Biostatistics, or CRAB, served as our independent statistical analysis team and our data aggregator. From a logistical perspective, we aligned on a statistical analysis plan as an entire group, and then the trial sponsors uploaded patient-level data to a secure portal. CRAB and the trial sponsors reviewed the data and shared private data set reports to ensure that the data that was included in our analysis matched what they had on their side. CRAB performed all of our analyses, and then we discussed findings and next steps with our project members in a nearly weekly cadence. And we aligned on key findings, and I'm here today to disseminate our results. So for research objective 1, we first defined the change in ctDNA metrics. We used max VAF, variant allele frequency, and focused on calculating a % change from baseline. Both of these were characteristics that were similar to step 1, so we were kind of trying to move on and use a similar approach from that. We did have a few clinical trials in this analysis that had multiple ctDNA values within the first 10 weeks, and for those we used the best or the lowest ctDNA to define T1 for patients. For molecular response, we had, like I mentioned, we looked at a change in ctDNA as a % change. We had ultimately 4 categories, but the 3 based off of % change were a decrease category with a greater than 50% decrease, our increase category, which had a greater than 20% increase, and then our intermediate category, which was between the 2. We noticed that in this data set, there were a lot of patients with non-detected ctDNA, and we had enough patients with non-detected ctDNA both at baseline and on treatment to create a separate category called the ND-ND category. Those patients who had non-detected ctDNA on treatment but detected ctDNA at baseline were considered a 100% decrease and were included in the decrease category. For statistical analyses, we created Kaplan-Meier plots, and we performed multivariable Cox proportional hazard models stratified by cohort, which included demographic and clinical confounders. We determined that categorizing the samples by % change did not demonstrate separation of the curves or statistically significant differences between molecular response categories. So we moved on to focus on detection, and we created 3 categories based off of whether ctDNA was detected or not at baseline and on treatment. The non-detected detected category had very few patients. It was an N of 6 out of our 1,000 patients, and so they were not included in future analyses. We see that patients with detected ctDNA at both baseline and on treatment had the worst associations with overall survival, regardless of whether their ctDNA increased or decreased. So we created this category of detected. We did perform a cut point analysis on this group, and we were able to identify a cut point that separates the data. Patients with non-detected ctDNA on treatment have a stronger association with improved survival compared with patients with detected levels of ctDNA on treatment. We moved forward with our molecular response category based on detection for the remainder of the analyses. So once we had our ctDNA categories, we decided to compare molecular and radiographic response. So our question here is whether patients who had non-detected ctDNA on treatment are correlating with our responders as defined by first RECIST. So within that first 10 weeks after index, we selected that time point specifically because it aligned with when our ctDNA values were collected as well, or a best overall response, which could have happened at any point during the study. And here I'm just showing that we, for both first RECIST and BRR measurements, we had very few patients that had CR or PD, and so we combined our complete responders with our partial responders into a responders category, and our stable disease and progressive disease into a non-responders category. When we compared molecular and radiographic response, that we see that across the three ctDNA categories for comparison with first RECIST on the left and comparison with BOR on the right, that the distribution of responders and non-responders across the molecular categories for first RECIST and BOR is similar. Our subsequent study is looking at research objective 3, focused on first RECIST because ctDNA on treatment time point was within 10 weeks, so we wanted to have a similar timing with the first RECIST. Also, BOR timing was variable and sometimes took many months, which meant that indexing would be challenging. So for research objective 3, when we were combining ctDNA and radiographic response, we performed 4 analyses. The first focused on radiographic response alone and associations with outcomes. So here we're not looking at changes in ctDNA at all. The next analysis combined radiographic response with changes in ctDNA to look at outcomes. And the third analysis was an opportunity for us to compare models to determine whether we whether we see improved associations with outcomes. Finally, we looked at molecular response within specific radiographic categories to see if ctDNA associated with outcomes within these categories, specifically looking at partial responders and our stable disease categories. For analysis 1, just looking at radiographic response, we see that there is no apparent separation between responders and non-responders at first RECIST and their associations with overall survival. For analysis 2, we created 6 categories combining molecular and radiographic response. The ND responders and non-responders were in blue. The DND responders and non-responders are in black and gray. And then the DD responders and non-responders are in red and pink. And while there are differences between the DD responders and non-responders, we also see that the molecular response categories continue to drive associations with long-term outcomes. Overall, we noticed that incorporating radiographic response at first RECIST into molecular response categories does not improve the degree of association with survival for those with non-detected ctDNA on treatment. And so then we moved into analysis 3, performing a statistical analysis about asking the question whether combining molecular and radiographic response improves associations with outcomes. We had 2 different models. Our full model was looking at changes in ctDNA with first RECIST and looking at associations with outcomes, and then looking at the reduced model, which is changes in ctDNA alone. And for our likelihood ratio test, we walked away from this analysis seeing that radiographic response at first RECIST does not provide additional value in characterizing the association with overall survival beyond the contribution of molecular response. And so we looked at molecular response within the partial response and stable disease categories, and we saw that the patterns were similar to when we looked at the overall data set. So, and specifically for patients that were classified as stable disease at first RECIST, molecular response does differentiate the associations with overall survival. And so our Module 1 key takeaways, first and foremost, we were able to harmonize data across 8 different clinical trials. It was feasible and we had an interesting outcome from it. In advanced non-small cell lung cancer treated with TKI, among patients who had detected ctDNA at baseline, patients with non-detected ctDNA on treatment, so our DND category, were associated with improved overall survival and progression survival compared to patients with detected levels of ctDNA on treatment. Molecular response associated with overall survival and progressive-free survival, while radiographic response at first RECIST did not. And among patients who are categorized as having stable disease at first RECIST, molecular response identified patients with improved associations with OS and PFS. So some strengths and limitations of our analysis. It was a very large data set. We had 8 clinical trials, 1,015 patients, 8 different TKI regimens. A strength of this is that we saw clear and unifying results across a wide range of features. A limitation is we didn't have an external validation data set. We saw variability. There were different assay types, different sampling timing for ctDNA and RECIST. So as a strength, our analyses were stratified by cohorts, so we feel confident in the data, and we had harmonization approaches that were used. A limitation here is that the potential differential in features due to variability despite the sensitivity analyses. We had patient-level data, which as a strength, multivariable models account for the patient-level confounders, and we were able to interrogate the ctDNA metrics. However, we were unable to answer some questions because of anonymization of the patient-level data. And finally, we looked at a retrospective analysis, which provided us with an opportunity to analyze multiple data sets with different approaches to data collection. And as a limitation, prospective analysis would be pre-specified. And so contextualizing the findings within our overall ctMoniTR project, Module 1 is not a validation of step 1, that smaller cohort of advanced non-small cell lung cancer treated with an anti-PD-L1. We had different treatments, TKI versus PD-L1, and we have different methods of defining change. We did a detection here, and in step 1, we looked at a % change. However, and I think most importantly, in both cases, molecular response associates with improved outcomes. And so Module 2 will focus on advanced non-small cell lung cancer with anti-PD-L1 or chemo, and it provides us with an opportunity to validate step 1. It also allows us to consider a different treatment. We'll have chemotherapy within that analysis, and there's a potential to analyze whether ctDNA can identify a more effective treatment in a randomized control trial. And with that, I want to thank all of our project partners. It is such a joy to be able to be on calls with you almost weekly and talk about all of these results. It's been a really great project, and we're looking forward to next steps. With that, I'll pass it over to Naveen. Thank you all so much.
Thank you,
So I hope that you enjoyed that very elegant presentation of what is just about 8 months' worth of work in, I think it was 15 minutes. So well done, Hillary. Also, a shout out before I introduce the panel to part of the team from CRAB. Anche Horing and Emily Gorin have traveled from Seattle to be here today. And so any truly difficult statistical questions will go to these 2 ladies, and they are here if you would like to discuss the data further. With my colleagues on the panel, we are going to try and unpack this for you, make the results come to life, and see where we go from here, and that will be a lead-in, of course, to the next session. So joining me on the stage, Karen Espenscheid from Guardant Health. She's a director in scientific affairs and also, I think, the external collaborations, and she's been part of our Module 2 work. We have Meenakshi Guha from Takeda. She is an associate director, statistician in the precision medicine group. Diana, who asks the everything questions. She was at Friends of Cancer Research at the very start of the journey, so pleasure to have her here now representing AZ. She's a director in the translational medicine group there. Paz Vilanki, who is a team leader at FDA, and she runs the team that looks after thoracic and head and neck interventions. Welcome, and you've been part of the journey as well with Module 2, really carefully dissecting the results as they come offline. And then Jeff Oxnard, who is currently Senior Vice President of Clinical Development at FMI, but was an academic and also at the table with us from the very start of ctMoniTR. So very pleased to share the stage with them today. So in terms of maybe the first question I'm going to throw out to the panel is, what's your takeaway? What is most striking about the results that you've seen? And Meenakshi, I'm going to throw it to you to get us started.
Sounds good. Thank you for the question, Naveen. And first of all, thank you for the invitation for Friends of Cancer Research for inviting us to this panel today. As we saw, Hillary did a great job in summarizing all of the conclusions. And what we saw is that the patients with non-detected ctDNA had improved outcomes, which was interesting on its own, because in the setting of non-small cell lung cancer and TKI treatment, it wasn't just the percent decrease or increase in ctDNA, but the detection or the lack thereof that associated with improved OS and PFS. And the other takeaway that I could point to is also very interesting is the fact that first RECIST alone did not associate with long-term outcomes, but the 3 ctDNA categories that we looked at did associate with improved outcomes, which was also very surprising, but an interesting takeaway.
Thank you, Meenakshi. Karen, I wonder if you want to continue us on the journey of those high-level insights.
Sure. Yes, for me, what was, I think, most surprising, at least at first, about these results was that when we looked initially at percent change for molecular response, the patients who had decreasing but still detected ctDNA didn't have a significant association with improved outcomes, as we've seen in previous studies. Now, that doesn't negate the previous results or the previous studies, but it does suggest that there are some other factors at play that we need to consider clinically and technically when we measure ctDNA change.
Great. And Jeff, I'm going to call on you, if you don't mind, to just make it practical for us in terms of these insights and what does it mean for future clinical trials?
It's worth recognizing we're studying a model system here, right? Mm-hmm. I'm a lung cancer doc. When I start a patient on targeted therapy, they're going to do well. This is a group of patients who do well on therapy. So in this model system, what have we learned? We've learned that ctDNA shows dramatic clearance. If you want to develop a drug that's like one of these drugs, you better be seeing ctDNA responses, right? But it's also not a setting of unpredictability. Like, we know how it's going to play out for these patients, right? This is very different than the first experience on immunotherapy where the therapy is unpredictable. In this setting, where we know what the outcome should look like, ctDNA trumps response, right? And actually, the thing we've known for so many years that the dreaded stable disease doesn't tell the whole story. It comes to life here, right? Stable disease adds nothing, and in fact, ctDNA can clarify a stable disease. So I think this model system allows us to now apply to other more mysterious therapies to really help amplify our drug development and figure out where are these drugs working and not working.
Great. So we've had some initial comments in terms of the insights, so maybe what was so surprising and then what we might do with it. We're going to take a bit of time to look at the details next, because that's I think, as scientists, we like to know the foundations are solid, and then we'll zoom back out and kind of go to the what's next. So one of the things we talked about is that there was variability in these studies that were, of course, created for other goals. And one of the big variabilities, I'm going to send this question to Diana, is the sampling time point for ctDNA. Hillary mentioned we came to this position of a 10-week window. Would you give us your views on this whole question of variability, the 10-week window?
Yes, certainly. First of all, thank you so much again to Friends of Cancer Research and the team to invite us and go over this module. That is, as what Jeff was mentioning, is a very clean model. You have an actual mutation that you're following, so that makes things a little bit easier. But as you were mentioning, Naveen, we have to keep our eyes fixed on what is it that we're trying to answer. And we had the data that we had. We had to choose a proper threshold for the timing window. I think that if the question is how early can we anticipate a benefit from a particular drug, it would be interesting to see it before maybe RECIST is normally measured in order to anticipate that and see whether a reduction in ctDNA level can anticipate RECIST response. Identifying exactly what is it that you're trying to answer, the intended uses was discussed on panel 1, is very important. The other thing that I'm going to pick up a little bit on is that combination with RECIST, also trying to figure out, are we using ctDNA to complement RECIST the other way around? Or in the particular case that we've seen here, should we look at RECIST in that particular setting the way we would see it in the step 1 setting when IO therapies are involved? So there's multiple nuances to take into consideration, but time points are important. As I said before, the baseline ideally needs to be done as close to when the treatment starts to be an actual baseline sample that you can compare with an on-treatment time point and identify the differences that are associated to the response to the therapy.
As we think of timing, I don't know if this is on, but Diana has gone through some of the considerations. I wonder if you would comment on, so what do we do with that for future trials?
Right. So I'll echo a few things that have been said already. I think it's really encouraging that we saw across multiple trials with multiple cohorts of patients that we were able to see very clear results. And we had, different ctDNA assays that were being used with different ctDNA time points. But I think as we start thinking about prospective trials and getting more robust data, it would be helpful to start thinking about how can we standardize some of these assessments? How can we standardize and harmonize ctDNA assays? How can we standardize ctDNA collection time points? That might show even stronger signals in future studies.
So when we come back to the question of, when we come back to the question of retrospective versus prospective, you'll probably pick up on that again, and you might all want to make notes to make sure that you ask those questions of what would be the value of a prospective exercise of this kind. So I just want to check with other panelists. I have another question that has to do with the foundations and the details, but any other comments on that question of timing?
I'll just say that some sponsors are tackling this. I see Vlad from Regeneron over here a moment ago. They just had data at ASCO looking at 3 weeks versus 9 weeks. I think you with your trials, please investigate this thing. Give us your feedback. With this data set, it's harder to cut it that many millions of ways, but we are learning. And it seems like week 3 is still kind of dynamic, maybe not fully settling out. Week 9 might be more of a nadir, but no question, we're I think, learning communally about this.
And then, sorry, I'm just going to add to that, and I think that it's context specific too. TKIs may work in a different way. You may have a different depth and duration of response that you need to evaluate with different time points compared to other assets or other drugs like IO therapies that may be, the duration is longer, maybe the depth initially is not as big, and that's why 50% was good enough for step 1, whereas we're looking at clearance in this particular module.
Right, and something we heard that was very important at the end of the morning session was, if we think of data elements and descriptors and data models, we need one of those for the assay characteristics. So in our Module 2 analysis, we really focused on understanding the impact of the limit of detection. We wanted to make sure we weren't seeing misclassifications when we went to that detected, non-detected that Hillary described. I wonder if there are other aspects of the assay characteristics that are relevant to discuss with the audience. And Meenakshi, I think I'm going to ask you to comment on it, but I'm sure there will be other points to be made by colleagues on the panel.
Sure. So we use ddPCR and NGS assays, and obviously we know that it has varying sensitivities and LODs, as well as different number of genes and types of genes that are assessed. So I think Friends of Cancer Research, the ctMoniTR project, did a great way of trying to be assay agnostic in the sense that we tried to harmonize, we tried to look at the change from baseline of ctDNA, which was a good start. And going forward, I think for other modalities and other indications, we need to start looking at various different assays, whether tumor-informed or tumor-naive, looking at perhaps a percent increase or a percent decrease threshold or cutoff may become important at that point. Also, there are assays out there that look at the counts of somatic mutations over time, over the journey of a patient, which may also make sense to look at. Currently, also in development, are assays looking at methylation patterns or tumor fraction, which can also be linked to molecular response. And I feel like harmonizing all these different aspects of ctDNA assays will certainly be very challenging, but one that we need to start considering now.
Thank you for that. Any other comments on the assay characteristics themselves, how they play into the work that we've done, but maybe also Meenakshi has hinted to future and other settings?
There was a comment earlier about the confounding of CHIP, right, and recognizing that if you're following a non-tumor signal, you're not going to have informative monitoring. And so just as we think about this, what does the next generation of assays do to contemplate filtering CHIP or algorithmically or white cell? What is the method so that you don't, your patient's responding, but you see a p53 that's flat, which is the winner there? And so as was mentioned before, that's going to need to be considered.
Okay, so CHIP is important and being really descriptive and granular as we share information. So assay developers in the room, bear that in mind. So we've talked about initial reactions. We've talked about some of the details that underpin the results. I'd like to switch gears and talk about tangible next steps. And as we think of next steps, we often want to contextualize. So Karan and then Diana, would you tell us how you view these results versus the step 1 results that Hillary mentioned very quickly?
Sure. So as Hillary began to describe towards the end of the presentation, the overall trend in step 2 here of ctMoniTR so far is similar to what we saw in step 1, which is that early ctDNA changes are predictive of long-term outcomes. But there are some apparent differences between the step 2 cohort and step 1 so far, many of which have already been mentioned, but a couple of the big ones are the patient cohort used in step 2 so far in this first module compared to step 1, as well as the metric used to measure ctDNA change. So of course, we in this cohort are looking at patients treated with TKIs rather than immunotherapy and looked at ctDNA detection rather than percent change. Now, for these clinical trials, patients were selected for enrollment based on the presence of a specific mutation that was targeted by that TKI. We would expect that that genomic mutation, that driver mutation, would be at or very near the max VAF in these patients if it's in fact driving the tumor. And we also typically see that these genomic drivers clear on treatment, as been mentioned a couple of times already. So especially in the setting where the patient is benefiting from therapy. And we also saw in this cohort that across all these patient trials, about 3/4 of the patients did actually have a partial or complete response to treatment. So this is what Jeff was getting at. Everybody did really well. So I think these factors of ctDNA change metrics and the treatment type really do seem to play together.
I mean, I think that step 2 was created to further try to convince us that what we were seeing in individual smaller studies, when you saw a reduction in ctDNA over time in association with a long-term benefit, whether we could extrapolate that to different settings. Step 1, we showed a 50% decrease was associated with long-term outcomes in an advanced non-small cell lung cancer setting treated with IO. Now we have another advanced non-small cell lung cancer setting treated with TKIs. And we have seen a reduction in ctDNA, perhaps different metrics, but a reduction in ctDNA that is considered significant is associated with long-term benefit. I think that as we keep looking at these studies, we are seeing that no matter how you look at it, 50%, 100% or clearance, detected, non-detected, ctDNA metrics, 10 weeks, once, twice, thrice, we are seeing that consistency in the message. Now comes the refinement and understanding whether the drug class is going to need some tweaking, differences here versus there, IO versus TKI. As was said before, step one was just a quick look. What is it that we have available? Can we see anything? We saw it. Okay, let's go into advanced non-small cell lung cancer setting treated with TKIs. Module 2 that is going to come in the near future is also going to be looking at that lung cancer population treated with IO plus chemo. So there's going to be another type of drug being involved there. So that refinement of understanding the metrics, the time points, is something that is going to continue evolving. But I hope everybody leaves this place saying, hey, we are seeing a consistency in that reductions in ctDNA are associated with long-term benefit. And that's what ctMoniTR was made to show.
Thank you for that. So we know that there's at least 2 instances where lower is better. We also saw something in Module 2, which was non-detected at baseline, also seemed to be a group of interest. We didn't dwell on it, but those who had non-detected ctDNA at baseline and remained such after the initiated therapy were associated with the very best survival outcomes. So now the question is, okay, what do we do? How do we incorporate this kind of insight that we have around the non-detected at baseline? And I'd like to, maybe Paz, I'll ask you to comment first, and then I'm going to go to Jeff and any others.
Sure. Yes, I think some of those findings are consistent with what has been seen with other analyses. Patients that don't have ctDNA detected at baseline have better prognoses. They have better long-term survival outcomes. And we saw that again here in Module 1. And so I think this again reinforces the idea that ctDNA can be used likely as a prognostic biomarker, and we should be thinking about how we use that in clinical trials. So for example, one thing that we could do, thank you. One possible way that we can utilize that is by using ctDNA as a stratification factor in clinical trials. We can enroll patients that are both ctDNA negative and positive, and we could look at outcomes for these patients. And we could also think about hierarchical testing procedures where we might look at outcomes in the intent to treat population, so patients that have both ctDNA that's positive at baseline and ctDNA that's negative at baseline. And then following that, you could look at results in just the group that's ctDNA positive. You also could consider doing the reverse, where you test the population of patients that's ctDNA positive first, followed by testing in the ITT population. And I think you'd have to think about the biological rationale for your testing procedure.
I was an imaging researcher before I was a ctDNA researcher, right? So I want to think about this in the context of RESIST. We've always limited our enrollment to patients with measurable disease, right? In lung cancer, that's 90% of patients have measurable disease and 10% are left out. In bone-heavy prostate and breast cancer, there are a lot of patients who don't have measurable disease who are omitted from some phase 2 studies, and maybe you study PSA instead. And so I think ctDNA maybe catches a slightly orthogonal population of patients, but could allow you to study patients who have less obviously measurable disease on imaging, but instead have measurable disease by ctDNA. I think it might vary by disease. I mean, in prostate cancer, this could be easier to measure than conventional RESIST. This could be your better endpoint, shall we say surrogate someday, I don't know what. But I do think contemplating how much of your population will be measurable or non-measurable, or maybe quantifiable or non-quantifiable, it has to be considered when you think about these tools in your clinical trials and allow you to design what is your IT population. But I agree with, the problem with RESIST is we ignore all those non-measurable patients in a lot of trials. So let's not do that with this. Let's make a plan for our non-ctDNA measurable patients, because they do great, and you don't want to omit them from your trial. If you're only enrolling ctDNA positives, you might be picking the worst patients. And so make a plan that considers the ctDNA negatives, tumor positives as an orthogonal population. We do have to design these together and not overlook this opportunity, I'd say.
That's a great point. I wonder if we could try and wrap a ribbon around this and talk about what comes next, either in the context of TKIs, advanced non-small cell lung cancer, or generally speaking, what do we do next, or how do we think about ctDNA as an early endpoint for drug development, large scale? I don't know, is there a panel member who might want to start us off on this? Because I'm going to come to all of you. Diana?
Okay. To end and put a ribbon on it, as you so
Like
nicely
ribbons.
said it, I think that we have to bring the patient, the reason why we're doing this, to the forefront. For patients to have more life-saving therapies, we need to, ensure that we have the right ITT population, and potentially is looking at ctDNA to ensure that more people are able to fit into different trials. Also thinking that the patient would really benefit from a drug that would benefit them in an accelerated approval manner. So identifying how early ctDNA, changes in ctDNA or molecular responses can help us move or expedite the development of drugs, because it's informing your decision earlier on instead of having to wait for long-term outcomes, instead of having to wait for confirm resist responses. Could you use this very powerful tool in your toolbox to help expedite the selection of drugs that move forward onto phase 3 IDs, or accelerated approvals, or any of these things in which the patient would benefit from. I think that's one of the things that I feel like puts a bow and a ribbon on top of this work, because that's the reason why we're doing this.
Thank you, Diana. Meenakshi?
I totally agree with that, and I think looking at ctDNA as an early endpoint or as an early marker of response is certainly more exciting and interesting to look at than first resist alone. And so we might start considering looking at ctDNA as a complementary biomarker to predict long-term responses. I think that would be a mind change that's necessary there, but I think it's an important one. And secondly, we don't need to wait years for OS and PFS readout to determine whether our drug is working or not. We can look much more earlier on. And in scenarios of accelerated drug approval, it perhaps makes sense to look at ctDNA as a predictor of response.
Okay, that's very bold.
Well, you use
We
the
like
word
bold.
excitement. All right? I want to think about those drugs you're excited about, right? If you think you've got the next alectinib or osimertinib, right, then how can you design a trial that meets your excitement, that is getting you data 3-6 weeks in that you are sharing with your investigators and investors or whoever? I'm not saying it replaces RESIST and you can wait 8, 12, 16 weeks plus 30 days for the CRFs to get the data back to you, whatever, if you want, and that'll come around. But in the meantime, design those studies where if you have a high concept study, this is a way to get that feedback quickly so you can accelerate your movement into registrational plans. So that's sort of the go, no-go window where it feels like this could really move our drugs towards patients more quickly.
And it increases confidence. I think that's, it's not one and only, it's all the different pieces, the accumulation of evidence to drive decisions. I think it's very powerful.
Very good. Other colleagues, Karen, you look like you're ready.
Diana and Meenakshi really, highlighted everything very well. I think there's not too much I would add, but I will say I'm excited about, potentially having more studies where we can begin to control for some of the factors that seem to be maybe creating a little bit of remaining variation in these results. And we certainly need more prospective studies that apply ctDNA along with the accepted endpoints, as was mentioned earlier today. I think that's really the next big piece to moving this forward.
Very good. And Paz, let's
And
hear from you.
Yeah, I'll kind of repeat a little bit of what Karen just mentioned. So we really do want prospective clinical trials because we're very excited about ctDNA as well, but we need more data still before we can use ctDNA endpoints for regulatory use. So this is, our call really to try to incorporate ctDNA endpoints into your trials. and incorporate ctDNA endpoints along with other efficacy endpoints so that we can look at the association between these two endpoints. We really need to get that prospective data. And then beyond that, I think, Dr. Cavazzoni already mentioned this, but we also would want to see meta-analyses. We want to see that across multiple trials, we're going to see the same type of association between the ctDNA endpoints and the long-term clinical outcomes. And so there are a lot of things that we need to think about in terms of what's suitable to pool for meta-analyses. And so this is where we need to have these ongoing conversations about, can we use data with ctDNA for patients that are being treated with IO? Is that the same for patients being treated with TKIs? Can we pool that data together? Do they need to be looked at separately? And so I think these are important questions that we need to continue to look at. And one final thing I'll say is that we always recommend for sponsors to come in and talk with the FDA. If you're going to incorporate ctDNA into your development programs, we want to talk with you. And we're excited about it, and we want to really capitalize on this.
Thank you for that. And one thing I would add in terms of the FDA interaction, there's fantastic coordination between CDRH, the senior levels, the review teams in the various tumor settings. And so I think Paz's invitation to come discuss with them what you might be doing to augment this body of work going forward is a very meaningful one. So I ran out of battery on my watch. I'm looking at a clock here. I just want to make sure it's the correct clock time because it is, very good. So we have time for your questions. So questions of any stripe, please feel free, come to the microphone, tell us who you are, and then we'll get you an answer or a reaction to the question.
My name is this on? My name is Kathy, yeah. Kathy Winston, I'm from Genentech. And I just want to follow on Pazdur, your comment, your ending comment there, when we're always encouraging people to come talk to the agency. Although when we come to the agency, we're talking about individual studies with individual divisions. And so maybe you can talk a little bit about what do you see FDA's role in, because we've also talked a lot about today about harmonization, standardization, and this effort that has been underway with friends, what, 3-4 years now? Yeah. We're pulling they're pulling together what they can get. And so we've talked a lot about, looking forward, how do we generate data that's more in line so when we do these meta-analysis. So what do you see as FDA's role in sort of helping that harmonization along? And is there some sort of work being done across the divisions so that we can encourage more standardization around time points and how we're tackling so the industry isn't going off and doing their own individual work?
Right, yeah, thanks for that question. So that's a great question. I would point out that FDA has been involved with a lot of efforts with ctDNA, including ctMoniTR. We've worked with BloodPAC. And so BloodPAC, for example, is having important conversations about data standardization. And so we're part of the conversations. I don't think we have all the answers either, but we're continuing to be a part of the conversations. And so, that's, yeah.
I think what you heard there was maybe a request if a sponsor does come in and say, hey, I'm wondering what my sampling schedule should be, it would be great to give them some very direct guidance, because otherwise they'll go off and do something that won't match the next person and the third person.
I'll follow up on that. And so, for example, a company could come in and could share some of their data where they've looked at different time points, and these are the outcomes that we've seen with ctDNA collection at 3 weeks versus 9 weeks, and we can have that conversation. So we are having those level of conversations with sponsors.
And if anything, this effort hopefully contributes to the learnings that will lead to the answers. If we're not asking the right questions, we're not going to get to the right answers, right? So if all of this, you think to yourself, well, what's the right time point? Well, then, guys, we got to work out the right time point. Like, we do need to make sure that we're evolving this program to address those questions. And, of course, if anyone's got data to donate, that's a really helpful way of moving us all in the right direction, and I know a lot of folks are working together to make that happen.
I'm going to come to the microphone. In terms of data to donate, if you are in a position and you want to join the collaboration, there are multiple colleagues from the Friends of Cancer Research Group that can help you with that. Yes, sir.
Greg Jones from NeoGenomics. Wondering if you can comment on
patients who radiographically show stable disease. There are some patients that have durable clinical responses and patients that don't have durable clinical responses that are considered to be stable disease. How does ctDNA play into this?
Meenakshi, do you want to take this one?
I think Jeff wants to take it first.
I'll speak to my clinical experience, please. You first.
Well, as we saw with some of the evidence that Friends of Research collected over here, this is the scenario where it makes most sense to look at ctDNA. And in fact, stable disease means it's stable. We can't tell if it's responding or it's not responding, and where does the patient, bifurcate it? And we can look at ctDNA as sort of an additional complementary biomarker to make more sense whether or not this patient is on a good trajectory or a poor trajectory. So yes.
Greg, I'll say in my clinical experience, some of those patients are progressing and you can't see it. And I do think ctDNA can find that. We've all seen that, right? Some of those patients just have non-shed indolent disease and your drug's doing nothing. And stable disease doesn't help you and ctDNA doesn't help you. And some of those patients are responding, and we've seen that in the clinical trials that have given you sort of bland waterfall plots and then given you ctDNA waterfall plots. It's like, whoa, there's patients in here that are benefiting. So I'm not saying that's a pervasive clinical problem. In the clinic, I'm meeting a patient, and they're like, I don't feel so good. I don't think it's working. I was like, all right, maybe we'll try something else. But in clinical trials where you can't ask the patient that question, getting this extra sort of much more sensitive tool, I think, could help clarify and could help some of those drugs that are in a sort of stuck spot to figure out if they're on the right track or not.
Yeah, Greg, we didn't do... This really doesn't like me, this microphone. Okay, we didn't do a subset analysis of the already subsetted stable disease population in the way you've described, but we do have an endpoint that's PFS6, which gets that durable response, and those results look very much like what you've seen already. Any other questions or reactions?
We have a few questions from online. One
Let's
of them
go.
has to do, kind of touched on it, everybody's really interested in LOD, but kind of assay harmonization, and do you think that we were able to harmonize despite we had different LODs? And we touched on it a little bit in the presentation, but maybe elaborate on that. And then the other question is about the stable disease data, demonstrating that we saw the same patterns there. What are the opportunities for using this data? Do we think that it can be used to better understand patients?
Okay, so I think the stable disease, we just had that conversation, but I'll check and see if the way the question was posed, there's more commentary.
I wonder if the data that we've seen today is going to get people to think of stable disease as, because right now it's kind of like a fuzzy in-between place. ctDNA, we've shown with this data here that it's able to determine those that have likely to benefit and they're considered stable disease. And even partial responders who are not likely to benefit long-term, you could also try to, refine that with ctDNA. So it's very clear in this data set, again, non-small cell lung cancer, advanced stage, treated with TKIs, ctDNA contributes that extra little bit of clarity that potentially is not there.
Okay, good. I'll follow up. I'll take the LOD question myself. So when we shifted to a detected, non-detected, you always have to worry with these varying LOD levels, are you misclassifying patients? And so what we opted to do was a serial analysis where we reclassified the next increment over into non-detected, and then the next one over, and then the next one over. We saw that in the first little piece where they're really close and conjoint, results did move. As we moved out, results did shift, telling us that we're not misclassified right at the joining point, which is the most important joining point. The other thing to bear in mind is that we stratify by cohort. So whatever assay differences live at the cohort level, which is where a lot of it lives, that's being accounted for in these results in the statistical multivariable models.
I'll say that I think detected, non-detected is the most vulnerable to variable LODs, right? And so
Variable LODs.
if assays have different LODs, they're going to have different detectabilities. And so that kind of dichotomous criteria risks being the most capricious when you try it with different assays, versus a 90% decrease is just like, well, kind of all the noise is just below the 90%. And so maybe we will learn some techniques to get around the randomness between assays. But I do think that there's going to be variability around assays that will be hard to overcome. You guys will not be the arbiters of which assays win or don't win. Like, so you just need to develop criteria or methodologies that might be more easily scalable across the different tools we use.
Yeah, and I would add to the assay developers, again, how you describe your LODs and the data that underpins that, make it public, please, because that's going to be very helpful to determining these methods that Jeff is pointing to. Other questions or comments? Anything online? Okay. So we're going to go ahead and close out. What I wanted to just reiterate is we had a small proof of concept with step 1 in the IO, advanced non-small cell lung cancer. We showed that you can harmonize, and we saw some very clear patterns. Those patterns, albeit with a different metric, seem to be reproduced here in the TKI setting, same cancer. We've got module 2 that's going to amplify what we saw in step 1. And then we have a mixed solid tumor bag coming with module 3. So we are slowly but surely going down the route of generating the insights and the knowledge to know precisely what role ctDNA can play in drug development and patient care. Thank you all. Jeff, back to you.
All right, thank you, Naveen. And thank you to all of the panelists and the working group members that have been moving this study forward for the last several months and will continue to do so, as Naveen just described. We have one more session coming up. I'll invite that group to please come grab a seat up here. And our final session today, the participants are going to help us think through the potential applications of this work, including additional perspectives on what's next in terms of operationalizing the use of ctDNA in drug development at various stages. I'll thank the group in advance. Come on up. For their comments and creativity, and look forward to the discussion. And I will hand things over to our moderator today, Dr. Amy McKee.
I don't think it matters. Rena's there. Oh. Okay, well, let's get started. It's an absolute pleasure to be here. Thank you to Friends of Cancer Research for advancing this important topic and moving along on the roadmap that you've created. It's incredibly important that we keep the progress going. And so with that, we're going to turn to talk about regulatory considerations for using ctDNA as an early endpoint. My name's Amy McKee. I am with ParXL. I'm an oncologist by training. Spent a lot of time at FDA, and I'm happy to be back here with some of my ex-FDA colleagues. Or they're still at FDA. I'm an ex-FDA. To my immediate left, we have Rena Phillip, who is the OCE Associate Director of Biomarkers and Precision Oncology at US FDA. Next, we have Nicole Gormley, who is the OCE Associate Director for Oncology Endpoint Development and the Division Director for the Division of Hematologic Malignancies 2. Next, we have Vladimir Yankovic, our Senior Director of Precision Medicine at Regeneron Pharmaceuticals. To his left, we have Moretta Liu, who is the Chief Medical Officer at Natera. Following that, we have Ken Billett, who's a patient advocate in numerous roles, including a mentor to patients with melanoma and a patient spokesperson frequently for pharma focus groups. And our last distinguished panelist is Christian Rolfe, who's a professor at the Icahn School of Medicine and the Associate Director for Clinical Research Center of Thoracic Oncology at the Tisch Cancer Institute for Mount Sinai Health System. So welcome to all our panelists. I hope we have a lively discussion, and we'll be doing questions at the end from the floor and online as well.
So I want to start with FDA since this is a regulatory discussion. As many of you probably know, FDA put out a draft guidance in 2022 about using ctDNA in regulatory decision-making in general for use as an early endpoint, and the importance of robust data and meta-analyses for validation were described within that guidance. So how are we doing? With the data that were presented today, how does this contribute to this effort? What other steps do we still have to make before we can start using ctDNA as an endpoint for regulatory decision-making, whether it's for go, no-go decisions to advance or even as an endpoint for marketing approval? And so for this first question, I'm going to turn to Nicole.
Great. Thank you very much for the invitation, and I'd like to thank Friends for the work that they've done here. I think it's really important that we have really wider stakeholder involvement in these efforts to develop endpoints. It's a really heavy lift, and when we come together and work together, we have much better outcomes than when we try to do this on our own alone. So that's the first statement. I would say I think it's also really important the data that was generated from these efforts. It really showed, as we discussed in the previous sessions, that ctDNA and its change detection, detected, not detected, that information that we can gather early on really does correlate with long-term outcomes. And I think what this underscores is its importance, as Paz mentioned earlier, as a prognostic biomarker. And I think in terms of how do we move this on and use this further in drug development, there's lots of uses, right, for this use of ctDNA, which, as we discussed, really has the potential to be a very powerful drug development tool. You can use ctDNA in theory for patient selection, for enrichment in clinical trials, as a patient stratification factor. You could consider use in clinical use as well in terms of decisions regarding treatment, treatment escalation, de-escalation, et cetera. And then obviously there's interest in using ctDNA as a clinical trial endpoint. And I think the next steps in terms of developing this particularly as an endpoint, and this was discussed earlier as well, there's really several pathways that have clearly been defined in terms of how to develop endpoints. The first being a meta-analysis using patient-level data, and then evaluating the association of the early endpoint with long-term outcomes on both a patient level, looking at the prognostic implications, and then also at the trial level, trying to determine whether or not you can predict the response to treatment based on this endpoint. And the other really common way is also just within an individual drug development program, prospectively collecting this information, and then correlating and seeing how this outcome corresponds or relates with other long-term outcomes of interest. And we often, in oncology, we talk about validated surrogates, we talk about surrogates reasonably likely to predict clinical benefit, we talk about intermediate clinical endpoints. The last two, intermediate clinical endpoints and surrogates reasonably likely to predict clinical benefit being used for accelerated approval. Most of our endpoints that we use, whether that be progression-free survival or event-free survival or even response rate, are intermediate clinical endpoints. But regardless, what we need to know how to use these endpoints and these tools in clinical trials, and that needs to be a data-driven decision. And so, we need to know if we're going to incorporate this into a clinical trial, what % change is clinically meaningful. What difference between treatment arms would predict that this therapy is an effective therapy? We need to know other aspects in terms of how to best define this, what are the appropriate thresholds, and all of those decisions, and when we're thinking about how to use this as an endpoint for a subsequent trial, need to be data-driven and data-informed based on our prior knowledge. So those are some of the outstanding questions that sort of exist right now in terms of how to get ctDNA to a point where we could use it potentially as an endpoint. I will say one last thing. I think, where there's a lot of excitement about ctDNA, and I think we're really, sort of in a great position here. But, we don't have to reinvent the wheel. This has been done before, so I think we can look at some of the examples of other endpoints and their path to development, whether that be use of MRD in ALL or the use of PathCR. And I think, some of the, and we can talk about this a little bit more, but some of the key takeaways are that, it really, a lot of these efforts really required a large group of people working together to do this lift together. And then I think, really using either meta-analyses or other other methods to sort of get the data that you need to be able to know how to use it going forward.
So you bring up some great points about how to integrate this into clinical trials. So I'd like to turn to Vladimir as a stakeholder who's building clinical trials and trying to incorporate ctDNA into them. Operationally, what are some of the considerations that you have to think about when you are using ctDNA as you develop a clinical trial?
Thank you, and again, thanks, friends, for organizing and for inviting us to discuss this important topic. So as pharma sponsors, we are in a unique position to contribute to this effort through the data we can generate in controlled trials, where we compare standard of care to novel therapies. And this is, I believe, the next step, right? After we develop some initial proof of concept in these meta-analyses at the patient level, now we can generate evidence at the study level, where we can develop the proof of concept that these early response measures, and I'm focusing on ctDNA here as an early measure of outcome, do translate in the effect size between the investigational and the standard of care therapy and long-term outcomes, right? So, but the question we have is, how do we best inform and how do we get the most value from these studies, right? So arguably the most valuable, other than getting the clinical efficacy data and advancing new therapies into the clinic, is what we can learn from these valuable patient samples that were collected on the study. And this is the greatest sort of capital that we have, right? So how do we best capitalize on it? So I would say a couple of things that, at least we're thinking about. One is, and this has been said already a number of times, is what is the best evidence for the required assay performance in order to have a meaningful outcome from these prospectively defined retrospective studies, which we can conduct right now. Meaning, how do we incorporate the relevant assays with relevant specificity, sensitivity? We talked about CHIP, we talked about LOD, right? I think this is some of the information that we can get out of the best data that is available right now. The next question then becomes, what are the most well-supported settings? And I want to go back to something that Diana said in the previous session, which I think is a very important consideration. Beyond the specific disease indication, how do we consider different treatment modalities, right? And what is the most promising near-term data that we can generate? Is this really comparing the different disparate treatment mechanisms, right? Where arguably there's a higher bar because of the difference in the depth of response, durability, and how they may translate into long-term outcomes, versus maybe identifying those consistent treatment mechanisms where we can reasonably expect better translation of the early response into the long-term outcomes. So those are some of the questions that we have. So again, assay, how do we, assay and testing, right? Time points and the right assay to include. And then where do we focus our efforts right now?
So to continue on that thought of, what is the right assay, what are the right thresholds, I'm going to turn to Rena and Minetta to ask you about what are the key aspects that you as a regulator use to evaluate regarding the validity and reliability of new early endpoints, And then as you develop these tests and incorporate them, are there specific criteria or thresholds for these ctDNA-based endpoints that need to be met to demonstrate clinical significance and regulatory approval?
Sure. First of all, thanks to Friends of Cancer Research for leading all these efforts, and I'm very excited to see the data that were presented today for both the baseline project as well as the module 2, module 1, step 2. So with regards to the use of assays for using, especially in this case, it's the use of ctDNA. What does FDA look into in terms of the device characteristics? One of the two things, as we heard this morning, sensitivity, specificity, and establishing good performance characteristics in terms of good sensitivity and specificity. It depends on what's the intended use, and I think the same thing we heard earlier. It depends on the context of use. So if you're using it for escalation, de-escalation, there are certain criteria that one would look into. For de-escalation, you're looking to have a test that has good NPV, good sensitivity, so you get less false negatives. Vice versa for the escalation. So similarly, for using a ctDNA as an early endpoint, I would the principle may be the same as in terms of when you're looking at the de-escalation. Maybe you should have a test with the good NPV, but you have to have that balance in terms of the performance characteristics. And we heard in the step 1, step 2, we have used different thresholds here. Step 1 use the percentage percentage decrease, whereas in step 2 we are using the detection. So what is the right threshold? Seems like we haven't come up with the right threshold, but I'm optimistic with all these efforts that are happening. We should be able to come up with what's the right threshold for a specific population. For the threshold that we established for lung cancer and TKI may not be the same as for lung cancer and for the IO project that we are going to look into next. But these efforts should come up with I'm hoping that's the first thing that we will be able to come up with, to have what's the ctDNA metric that one should be looking into, and what is the right threshold one should be looking into for a specific population. It definitely will vary with the population. And when we talk about the threshold, we should also talk about the timing, which we heard this morning. The timing definitely is something that we should be looking into for a specific population.
Thank you. Thanks, Amy, and thanks, everybody, and Friends of Cancer Research for inviting me to serve on this really esteemed panel. One thing, just to take a step back, we have to remember what we're trying to answer. So ctDNA has the ability to help us in multiple aspects of clinical trials and patient care, right? It can help us with getting risk stratification, enriching your patient population, escalation, de-escalation. We're here really to focus on treatment response monitoring and the use of ctDNA as an early endpoint. And so does that mean, so it's fit for purpose as you're developing an assay or you have an assay in hand as a diagnostic company, where are we going to fit best? And then where you fit, then comes the questions of thresholds, et cetera. With respect to using ctDNA as an early endpoint, first, as we've just seen with the ctMoniTR project, is it clearance, or are there increases or decreases? And that's a really important question. Is clearance all we're going to achieve? Is that really sort of that black and white definition? Is that really going to help us over-resist? Are we going to get a better understanding with ctDNA kinetics, where quantitation really can be helpful? And that's where the time point, we've heard this theme, right? Depending on the drug, depending on the tumor type, depending on the stage, the kinetics of ctDNA are going to vary. They're probably going to vary by assay to some degree as well. So it's really taking advantage as those in drug development are conducting clinical trials to collect the specimens that are needed. And unfortunately, it's that investment in multiple time points to really help us understand. If we have so much rigor that we say do it, at week 3 and week 9 always, we're going to miss out between weeks 3 and 9. And again, different drugs are going to mean different things. So that's the huge investment. And I don't have an answer about thresholds because I think we really need to define that. And those thresholds, again, may vary. It's clear ctDNA is going to help us. Right? It has so much potential. That's what we keep hearing. Now it's incumbent on all of us to help understand how to truly implement ctDNA in practice and perhaps more importantly in clinical trials for drug development.
Yeah, I want to return to something you said at some point later in questions about continuous versus a binary endpoint, because I think that's a very interesting point for oncology endpoints in general. But maybe that brings us to our next thought about what are we going to do in the next trials. We've been doing some prospective, retrospective looks at studies that have already been conducted, but what about future trials where we're planning them right now? So how are we going to continue to incorporate ctDNA into planned studies? And so Christian, I'd like to turn to you for this question first.
Yeah, thank you for the invitation. And so I think here we need to consider different things. First of all, we was discussing before during all the day technology, assays, kind of designs that we are using. But particularly we are sometimes not incorporating liquid biopsy as a final tool. We are putting only as an exploratory analysis, and that has happened several times in different trials, not only from companies, but also from organizations or cooperative groups. So that is very important, and also the idea that we can include in the future populations and arrive at populations that we are not really including them in the clinical trials. So, for example, also the kind of model that we are following in drugs now with the pragmatic approach to try to incorporate these populations, relaxing a little bit sometimes the inclusion criteria to try to get this data. Validation or retrospective analysis are really important, but also here we have a very difficult situation because in some, if we are taking, for example, the TMB, is one of the easy examples to take that here we was not doing a great job, so we was putting together different assays, different thresholds, different technologies, so it was really very difficult to get a conclusion there. And that is my afraid I would say on ctDNA dynamics also, that we are doing different trials, that we are not really putting together efforts to get a unique scenario with the only technology. So it's very difficult to get answers from different points. So in the terms of designs also, we need to see the feasibility that we have to apply the assays in the future in different clinical trials, and will depend obviously on different aspects. If we are talking about minimal residual disease, if we are talking about early detection, so we need to be very careful with the scenarios that we are applying to try to figure out if sensitivity and specificity are playing the same role. And here we need the statisticians, especially for these dynamics, to try to get a conclusion which one is the best endpoint to arrive there and get the power of the conclusion.
So Vladimir, I'll turn to you. How are you thinking of this from the sponsor perspective in
so
future trials?
going a little bit to what I was commenting on before, so I think there's maybe different levels of prospective trial design that we can consider, and those require different levels of preliminary evidence. So something we can hopefully do right now based on the information we can get from the current data, from the meta-analysis, such as the one that's being conducted here by Friends of Cancer, is to prospectively define these endpoints and include them as exploratory analyses. They will be prospectively defined, but they may not be possible to include as part of the statistical hierarchy with specific statistical hypotheses. And the reason is we don't know what the effect size is. We don't know how to power these right now, and it would be very difficult to justify, alpha spend, for instance, for such unknown, outcomes. And this is basically the way we think about this. It's going to be a stepwise approach. So currently what we're thinking about is really this prospective, retrospective design, where the value we're bringing is that we implement the best assay approach, the best time course, select the best clinical setting, early, late line disease, answer the key questions that are really best supported by the existing data. And then that will then inform, once we know what effect size how the effect size, the ctDNA translates into the effect size in the PFS and OS, right? That then we can, be informed in how we power our statistically designed prospective studies. And again, to the previous point, there's precedence for this, right? So, there's a lot of precedent that we can follow in how this is going to be done. So that's basically my main point, is that there's going to be a stepwise approach in this prospective design, right?
So what I hear you saying is, as excited as we all are, your next study still will remain an exploratory endpoint so that you can understand the assumptions in the exact disease context and exact type of therapy before you can pull it into a hierarchical.
This is my opinion, right? That realistically, this is where we need to start. Doesn't mean that we're not going to do this in parallel as well. Maybe there are some settings where we can, be informed already right now. But I think those are at least two different levels of evidence that we can generate.
And Christian, I think you had a
so coming back to the point of retrospective analysis, I think here we need to do a major effort between academia, companies, all together to try to get all the data that we have. There is a bunch of data coming from different trials that are still in the company side, or data that we have in cooperative groups, that we can extrapolate this data and try to validate new tools. For example, if we are talking about tumor fraction, for example, it's something that we can recuperate this data, try to validate in retrospective analysis, and learning from there, go to prospective clinical trials. So I think it's a missing opportunity to don't take this data that we have already storage in some place.
Yeah. I don't know if there's time for just one more comment here, is that one other approach that we've been thinking about is, how do we advance the technologies as well? So if we do design, say, prospective small study where we implement the current assay technology, right? Is there an opportunity to collect a parallel sample set that will then be used to retrospectively test in a setting where we already have a benchmark, right? So is there something pushing the field along, and working with academic investigators is a big opportunity that we're considering in medical studies.
So Nicole, I'll come back to you. What are some things that you would caution as pitfalls in the next study design, and things that you would want to make sure people are including in the next study design?
Yeah, I think, one of the big considerations here is just, and I would really encourage, as the field, to try to work on standardization, right? I think, if we could sort of have some degree of standardization in terms of the times that we're assessing this, the thresholds, et cetera, it really can help then just advance the field altogether. When you think about trying to go back and pull this information or data from a retrospective, meta-analysis or in some other manner, if there's so much variability in the times when it was assessed, or so much variability in the assay, it sort of limits your ability to draw meaningful conclusions from that data. So, to the extent that there can be more standardization across the field, it really does, I think, have the potential to, sort of rise all boats, as we were talking about before. And then along with that, then, I think, just as you were saying, even if there's not comfort in having it, as a secondary endpoint with appropriate alpha allocation, still making sure that that's assessed, in clinical trials across the board, such that then you actually have more data that you can evaluate and answer questions for how to subsequently use this. So I think those are the points I would make.
Thank you. Ken, I want to come to you next. As we're talking about the next clinical trials and how to design them, let's take it from a patient perspective. How do we build these trials that we get meaningful data out of them, but it's not a bigger burden to the patients who enroll in them?
Right, which should always be a consideration, is what can you do for the patients? What can you do to make their journey a little bit easier and a little more stable, if you will, make their world a little more stable. I wanted to mention what Amy said. I'm a current stage 4 cancer patient, and I'm also, to the last panel, a poster child for stable disease. And so I'm a person who definitely would benefit from this technology moving forward. And I think that's one of the things that's very important, is making sure that we lessen that burden, we somehow personalize that interaction between the researchers and the clinicians and those patient participants. And I think ctDNA gives us that opportunity. Something that is as simple as taking a blood test and being able to use that to help augment regular CT scans, for example, or a PET scan, not replacing, but actually being part of that process. Anything that makes the lives of the patient participants easier is going to be a huge benefit. And I think that's probably the key thing. Also, I think it's also, on a certain level, easier for folks to understand. Patients, caregivers, families, saying that you're looking at blood, you're looking at what's going on with the tumor and being able to evaluate that. I know I'm simplifying it, but I think it is a way to do that that helps everyone and eases the burden, gives people a reason to want to participate. Part of what you mentioned too with my work that I do is that is one thing that we've seen is access to trials as well as understanding what the benefits are of that. So from, again, a patient advocate perspective, that's very important.
Thank you. Christian, as the person who's in the room with the patient talking about enrolling in trials, how do you view this about trying to make ctDNA part of the studies, but not making it overly burdensome and still meaningful?
Yeah, no, that's a very good question, and I think it's coming back what Ken was telling right now, is try to figure out what are the gaps and the needs from the patient. So sometimes we are seeing several trials that are Me Too trials, so they are continue to be the same proposals. So we need to try to get out from these triangles of fashion, market, and try to get more in what the patient needs. And sometimes questions are not really are very scientifically appealing, but are not so marketing appealing. So that is our commitment as academia to try to answer that question. So and get support for answer that questions and try to be innovative in clinical trials. So I think this is something that we need to work together with companies, patients, advocates, and academia, and try to get these answers because it's really needed.
All right, so let's continue talking about next steps. Rena, this question is for you. What are the challenges in implementing standards and methods in this rapidly evolving field? I mean, we've heard about a lot of different methodologies. Things are changing all the time. What is your recommendation?
Yeah, so we all acknowledge that the methods that are used to assess ctDNA changes, they are rapidly evolving. They are, be it tumor-informed, tumor-naive, now there's plasma-informed. So there are, lots of these different innovative new technologies that are rapidly happening. And there is significant variability in how the cutoffs are defined. There are, maybe some complex statistical models are there. But, yes, we understand this is all happening. But we have to do meta-analysis, which is what we are talking about. We need to have some, clinically valid definition of what is actually considered as MRD negative. And that has to be decided in the context of the tumor type. That's what we were discussing, tumor type, the drug, and also the stage of the disease. And so, before we go into the next, maybe the next step 2 of, we have to think of that in terms of what are we going to look into? What is the definition of that before we pool all that data? Because, even for the baseline, the way I interpreted, yes, they are kind of comparable, but there are big differences the way I saw it. So that could be because of how these assays are, how they're measuring it. It's how the ctDNA is measured with different assays. So how, even if, let's say, we are looking at delta ctDNA, are the delta ctDNA between these assays are comparable? Can we at least look into that before we actually, really look into 10,000 sample data, because that's, then it makes sense, right? I think I've said this in the past, and Dr. Pathak also mentioned, there is, the reference materials, standards, maybe, and I know if NIH is working on it, I don't know whether they're ready to compare between the assays, but that's something we could actually maybe do, maybe as the first step, and see whether these assays are comparable, or is their LODs are comparable enough to make sure that, whatever the threshold we are coming up with is, it's going to be comparable between the assays, because then actually, when you're pooling it, the data will really make good sense of it.
Nicole, I'm going to turn to you next, because I know a lot of these issues came up with some of the other early endpoints that the oncology group has used, such as Patrizia, and I know you were intimately involved in minimal residual disease in the guidance and some actions based on that. So what are some lessons learned from that you would apply here going forward?
Yeah, I think, it's true, right? We're always improving and getting better, and it's almost always like a changing goalpost a little bit. But in some ways, I think, it's that's great, but, it's also really helpful, I think. What you can do then is just sort of like, from a data standpoint, identify what threshold is actually clinically meaningful. So like in MRD, for example, there were a lot in multiple myeloma. There were a lot of evaluations looking at what best correlates with long-term outcomes. And, it was ultimately decided, 10 to the negative minus 5 actually might correlate best with long-term outcomes. Well, our assays improved, and then we're looking at 10 to the minus 6. But you still have that confidence that at least at, anything at 10 to the minus 5 or less still would correlate with long-term outcomes. And so I think that sort of approach, and it sort of mirrors what we're talking about in the oncology diagnostic pilot as well, sort of developing, what are the key thresholds for the analytical performance that if you at least attain this threshold, that would correspond or correlate with long-term clinical benefit. I think you do have to be careful though, right? So, deeper responses are not always better. And sometimes if if that's at the expense of safety, you may still lose that correlation with long-term outcomes. And so in many of the clinical trial designs that we talk about where we use these early endpoints, we still continue to follow those patients for later long-term outcomes. And you sort of really still need both to ensure that the long-term clinical benefit is actually there. And so, we use early endpoints, and we think that they're really, really helpful to expedite drug development, but we still continue to follow those patients for the long-term outcomes to make sure that the clinical benefit that we expect from those early endpoints is actually there. And then just as a last plug, that's sort of, again, where it helps to come in and meet with us in the specific drug review division to ensure that your clinical trial design is appropriate to sort of help make sure that we're getting the answers that we need.
So I'm going to move to a very general question that I'd like each of you to answer, and maybe I'll start with Ken this time. How can we get this field to continue moving? What are you doing? What do you want to see done? How can we keep going and pushing on this roadmap?
My original answer to that was education. And by that, I mean educating patient organizations like those that I represent, advocacy organizations, the general population about the benefits of ctDNA and the technology that's there. Also getting them to understand that we're here and we need to be here and we need to keep moving forward, and that participation in clinical trials helps that process move forward. So it becomes a more reliable, more measurable benefit to everyone. And then also lobbying for funding, lobbying the FDA, for example, and on all the stakeholders. I work with ctMRP to kind of get off on a subject, so I'm kind of used to this whole thing, being a the consumer reviewer as well as one of those people who goes to the Hill and talks, to well, not to the Congress people, but to their to their aides and so on. So yeah, I think that's the key thing is education from my perspective, is getting that out, getting the word out.
Okay. Minetta.
To move the, I mean, I think we're doing that here, right? Having representation from FDA, pharma, diagnostics, advocacy, and importantly, academics, having everybody together and just collaborating around, we're creating a field together, which I think is remarkable. But along those lines, we actually have to act together. So there are important efforts to create lexicons, like just even identifying like what the vocabulary is. It's not that easy. You could be talking to people in the same space using the same words but meaning something different, and that's not helping us. So that degree of collaboration. And I think it's also the opportunity for efficiencies. You were talking about, from the patient perspective, facilitating clinical trials. We have the ability now in their movements to decentralize clinical trials, the ability to use a blood test where you could do mobile phlebotomy, where you can draw the blood, in a private healthcare provider's office, that the patient can be evaluated there, right? Those are efficiencies, not just importantly for the patient, but also for the clinical trials. Not enough patients go on studies, right? And clinical trials really should be part of the standard of care. So we should behave that way, and it should be able to be done at home, not having someone sort of transfer themselves. And then the other efficiencies are in how we operate and how we interpret the data. We're talking about early endpoints, all relative to clinical outcomes and survival, but there are other benefits as well that we have to think about just in terms of health-related quality of life, the finances for a patient as an individual, for us as we're all trying to move this space forward. So those are all important considerations as we're trying to develop ctDNA.
So I started in the middle of the row. Maybe I'll go back to the end.
Yeah, so I think education, standardization, globalization of the concept. So from this is a very, very innovative and very dynamic field, and we are the same people working in the same field. So we need new figures, for example, working in liquid biopsy, and that is involving not only doctors and patient educations, but also, for example, including nurse practitioners, nurses, becoming more molecular nurses, trying to create career ladders for this healthcare team to try to improve the access. Globalization, and we are doing that, for example, from the International Society of Liquid Biopsy. We create a specific pathway for this part of the team in healthcare, and I think it's very important. Globalization, we are talking today and we focus our discussions on US, but if we are talking about the worldwide, there are very important disparities in the access to the technology, in the access to the application of this. So I think we need to work a little bit more global. And I just back, for example, from South America in a congress, and we was discussing about liquid biopsy, and we are still discussing and the access and the opportunity for patients to be tested in a single gene. So it's something that is really unbelievable, the difference that we are living worldwide. So having the access and also the opportunity to educate also not only doctors and patients, but also sometimes in different countries, regulatory agencies that are not, we are not outside US so lucky to have FDA always. So it's very important that we also invest some time also in worldwide application.
So that's actually a great way to pivot to you, Vladimir, because I'd love to hear your answer, but also throw a little bit of a curveball question. How in global studies are you seeing different levels of sophistication and ability to look at ctDNA to Christian?
Yeah, so I'll answer that first. So I think that's a very important question. I think it is, now much easier to implement the proper sample collection with some of the novel technologies, upstream sample processing that allows us, robust sample collection across global studies. What I do see is an important limitation and something we need to consider, especially for to the extent that we'll find that tissue-informed assays are critical, is the tissue collection, right? And so this is where we, anticipate a lot of challenges and need to take a very specific approach to different geographies and how the samples are collected, available, what is the standard for sample collection, what is the quality of those samples, and that will impact the rate of data generation that we can expect, especially from these large studies. So that's what I can say about that. But I did want to go back to your kind of closing question and use that to give another pharma perspective, and something that was alluded to by my pharma colleague this morning is there's an important synergy as we are developing these early data right now with some of the information that we can get for our internal decision making during development, beyond and above what we expect later on to be a regulatory approval. So I'll just give 2 examples, just kind of illustrate how we're thinking about this. This potential to adjudicate ambiguous radiographic responses, refine exposure response analyses, maybe gives us another opportunity to optimize the dose, right? There's a lot of initiative to optimize the dose selection in oncology, Optimus project, right? So, and this is, I think, an area where, in addition to kind of aligning on the long-term goal, right, it will be very helpful to standardize and align on what are those opportunities within development, even if not for regulatory approvals. How do we view, enhanced response assessment for dose selection? Another one is, can we further, as we're moving systemic therapies early into perioperative setting, right? Can we use MRD clearance to understand whether adjuvant therapy is needed where you have perioperative, neoadjuvant, adjuvant treatment paradigm? So some of these unanswered questions that, even though they may not inform the regulatory approvals immediately, I think are very important to consider and standardize across, pharma, and get input from academia, from other stakeholders, how we implement and how we use those data for our internal decisions. So I think that's important.
So just pushing drug development forward as well as clinical care.
Exactly, right, because that is what we ultimately have to make our priority, bringing these patients to, these therapies to patients sooner, at within a shorter time frame, and with smaller data, with smaller study sizes, right? So if we increase the power, for instance, for detecting, early responses with more certainty, defining optimal dose sooner with smaller, defining the proof of concept, understanding which therapies to push forward, that will be a very important incentive for our industry to, generate more data that can then also be leveraged for the long-term goal.
Great. All right, Nicole.
Yeah, I think I would just echo what's already all been said. I think, standardization is absolutely key to try to, make sure we collect the same information, and then that improves the strength of our analyses later. And then I think to do just that, standardization, and to move the field forward, there has to be collaboration. And so again, I really applaud Friends and their effort here, and just encourage people to continue that, because I feel like to move this field forward, that's what it's going to take.
And Renee, you get the final word before some question and answer.
I would say data generation, more data generation, because now we have seen the potential of ctDNA as a prognostic biomarker. We need to incorporate this into clinical trials now and get more data. And prospectively controlled trials, that's important, that's needed, right? And come and talk to the respective divisions in terms of getting their advice on how the timing and all the details for the drug development. So meta-analysis is important, as well as prospectively controlled trials.
Great, thank you. So That's the end of our formal questions of the panel, but if we have any questions from the audience or from online, please come to the microphone, introduce yourself and your affiliation, and then you can ask your question. While we wait for any questions, I want to return a little bit to measuring ctDNA and whether it's a continuous or a binary endpoint, because I think we use mostly binary endpoints. We've tried to use volumetric assessment, and that's never really quite taken, and maybe because we haven't put enough effort into developing it as an endpoint. But where does everyone think this is going to go? What do you think is the best answer in this space? And Vladimir, I think you want to answer.
I'd like to address this just based on some biology insights that we're starting to get right now. At least from my view, ctDNA responses may be bimodal, right? So they tend to not be continuous variables, and sometimes it may not be, there may not be a difference between looking at no decrease, any decrease, 90% decrease, or even clearance, right? So this is just one thing to consider, just the biological properties of ctDNA changes that we're starting to appreciate. So that's my view, is that it may be maybe not a binary, but maybe a very, very bimodal distribution of these responses. So
I think Christian has a response.
Yeah, I think one of the problems that we will have for the continuum will be when we are applying liquid biopsy in the design of escalation or de-escalation clinical trials. So that will be very difficult because it depends on several biological components, but also from the point of view of the statistical analysis, it will be very difficult to find a threshold there to use the liquid biopsy as a tool of the decision-making in a clinical trial.
Great. I see we have some questions from the floor.
Hi, Raluca Bradoio from AbbVie. Great discussions here and a lot of learning from my side this afternoon and this morning. I heard from, I think, every single one of the panelists the word standardization. And I think everyone agrees, and myself 100%, that is needed. But I wonder if there are already plans to start something like that, and who should drive something if there's nothing started yet? And how we can tackle that as a field?
Maybe I can, I know BloodPack is actually having some global regular discussions. So maybe that'll start there in terms of the standardization of collecting data elements. Maybe someone in BloodPack is here who can say more to this.
So no, minute.
Was you?
No, I mean, there's standardization in the definitions, like we talked about before. So the lexicon, standardization of data collection, of timing of collections, of blood collection tubes. How are we going to simplify this for the users, if you will? And then the standardization from the laboratory perspective. And as we keep talking about thresholds, I keep thinking, threshold for what? Like the threshold for detection. Is there the threshold above, the percent change for increase or decrease? So all of these things, I think, have to be defined. So there's at least a recognition that it has to happen. There are remarkably groups that are pulling folks everywhere. FNIH is doing this as well, creating reference standard materials so that we have some level of control across different assays. So all of those components are there. I think they just have to get pulled together like everything else.
Just one to add to this. I think analytical standardization may not be as difficult, right? It's something that we can, there's precedence for it, we can think about it, but I think we first need to start on what is clinically meaningful, to your point, right? What are we trying to standardize for? So I think this is maybe the first step, understanding what type of assay performance we need, right? And then we can design the appropriate studies to standardize to that standard, so.
Yeah, and also from the point of view of standardization outside the vendors, so outside the commercial platform, we need to also look what is happening in in-house technology. So we are trying also from the Society of Liquid Biopsy create a standardization, guidelines for minimal requirements to work in liquid biopsy in labs. because we see that there are several differences in the labs, and sometimes we are using in different regions in the world, they are using that for decision making in patients, and also using outside ctDNA. In some cases, we saw some CTCs or exosomes data, microRNAs that are there, so we need to be very careful to protect the patients also from that point of view.
Jeff Oxnard, Foundation Medicine. I want to posit that some of these things are all very standardizable, right? And others are not. There are methylomic assays and tumor fraction assays and fractomic assays and tumor forwarding, like they're just not standard. The technologies are nonetheless diverse, and Reen, you spoke to this. It's like, I think you kind of said, stop blending all the assays together if you don't mind. And I think, the part of the goal here is to describe a biological phenomenon, and I think we're doing that, and you're like, thanks for describing a biological phenomenon. Now tell me which assays can do it. And that's not really the role of this body. And so I guess, which body is it, by the way? Is that what these little confidential conversations with the FDA are about? The sponsor brings and says, our data looks like this, has that look, and you let them know? Is that on the sponsor to validate their assay? Are you assessing each of the assays in your new role, Rena, to decide if they meet this? Like, who is the person who decides that an assay can be used for Vlad's dose-finding experiment, right? Where does that validity experiment happen? Because I'm not sure it's clearly laid out, because back to the point, I'm not sure they're all standard. But some of them can do this, and how do we know which ones?
Maybe Nicole has the MRD experience with the,
Yeah, you're asking there's two things to keep, and you touched on this. The first is that, we talk about the analytical validation, and that's whole, component that we collaborate with, CDRH on and have those discussions. But then we talk about what's the clinical validity, separately, like how is what we're, the results and the difference that we're seeing, is this clinically meaningful, and does that correspond with whatever outcome we're interested in? And those are two separate, discussions. And so, there's various ways to go about this, and it again sort of depends on what you're planning on using the assay for. It depends on the context of use, very much so. So, if you're talking about this as an endpoint, and if there is some standardization, then you can, as a group, it doesn't have to be an individual sponsor, right? As a group, there can be collaborative groups that come in and say, hey, we want to validate or discuss the clinical validity of this assay, endpoint, et cetera, for drug development as an endpoint. And that, is a discussion that can occur separate and apart from a specific drug development program. But there's that, but again, it really boils down to what you're talking about using the drug for, or the assay for, and the endpoint in drug development.
To follow up on that, I think, to what some of Vlad was discussing, that's an individual company's decision. Like, if they're using it for proof of concept, they need to determine what thresholds and what assay and the validity of it, because that's not an FDA issue.
Exactly.
That's a drug development company sponsor issue. Where FDA comes in is when you need a decision on something for your drug development program. So we're really talking about endpoints, about diagnosis, about response, all of these things that FDA has some purview over, which is a completely potentially different, maybe overlapping, maybe Venn diagram. And then there's clinical care, which also is part of that Venn diagram. So I think we just have to be careful about, to Nicole's point, exactly what is the intended use for the assay.
The specific one Vlad mentioned was Optimus dose finding. Okay, that's a shared one. So it's like, we use the following information as we chose our dose. What do you think?
And from an FDA perspective, like, we look at the totality of data, and if you're using an assay that's not analytically valid, we're not going to really consider that very much in terms of the appropriateness for the dose. I mean, but that's not to say that there's not, other information, or it can't be a part of it. But again, that's where it really works to come in and talk with us beforehand.
Just to add, what I was really referring to the industry alignment, industry vendor alignments, and these types of things, exactly to the point. Let's get the right assays. Let's generate the most robust data, which we can then present. That may not be the FDA.
I want to hear what Ken thinks about this conversation as the patient on the panel, because you hear all of us talking about someone has to do it. Who do you think should do it?
Oh, who do
who do you want to put, like, you should do this?
Me personally should?
No. I want you to tell us who you think should be responsible for this.
I'm going to have to take a pass on it.
Okay, fair
I'm
enough,
going to be diplomatic.
fair enough.
No, but I have a comment. So what we need to be careful also is that several companies are going directly to the market and with, yes, very nice data. For example, if we take in the case of early detection, very nice data, but is ready to be applicable around the country, or is able to be easy with the data that we have applicable? I was recently in a congress for screening, and I was asking how many of you in general oncologists are using different tools that are available commercially, and several general oncologists are raising their hand. So we need to be very careful also with that, and very, I would say, responsible in the decision and the education that we are giving about the technologies, because it's very exciting. We are very happy that we have technologies, but what is the real meaning of all this applicable in the clinical practice?
Thanks.
Hi, Naveen Zarifa, NMD Group. So I find it hard not to be humble in this kind of setting, in this conversation. I'm thinking through the dimensions that we're trying to balance. We've got patients who require new medicines and a throughput from R&D that's better and clearer than what we've had to date. We've got smart technologists and scientists creating the new version of the assay before the previous version is like understood. You've got these large collaborative groups that are going, hey, wait a minute, I think there's some promise here. And I'm wondering, what's that inflection point where we say, let's put the guardrails down and the anchor points, answer this question with that, we will achieve this outcome. Because otherwise we make it more complex and we get lost in the breadth and the depth of all of those other dimensions before we're on solid ground. And so I wondered if there was one imperative that you'd be able to say, that will move the needle for ctDNA, which in turn moves the needle for R&D throughput, which in turn moves the needle for patients. What's that one imperative? Top of mind, not all the complexities, the one core thing you would say, don't leave this room without a plan to do X.
I would say valid definition of the MRD negativity cutoff for specific context.
So
the first thing.
Right, so across different contexts, different drug classes, what's that cut point? Yeah, that's clear. Thank you.
Were there other responses to that? Were there other, I mean, I've heard standardization.
Yeah, I would put that in there. But it all sort of works together, you know? Like, I think these are all that we're all saying the same thing. And I agree, like, I think it's a stepwise approach, you know, that's really needed to move this forward. That, you know, you need to get more data, and it needs to be somewhat similar data, so that way you can actually analyze it and use it later. And that requires everyone working together to sort of have those next steps. So I guess I would just really underscore that when you're thinking about using any endpoint for drug development, and especially as an endpoint to determine the safety and efficacy of a therapeutic, it really needs to be a data-informed decision. And so however you get that data, hopefully by working together and having, you know, standardized approach, I think that's the best, you know, way, but you need the data.
In my opinion, next inflection point after we have developed a proof of concept is the proof of concept that we can actually compare different therapies, and we can have meaningful assessment of early differences in therapeutic outcomes with investigational therapies. So that, I think, is the next inflection point that we as pharma can be thinking about.
All right, we may have time for one last question if there are any questions online. Any other questions from the floor? Okay, thank you very much to the panel. Thank you again to Friends of Cancer Research.
That was really good.
All right, we'll just make do. Okay, thank you. Thanks everyone for joining the meeting today. I think we recognize there's a lot of work to be done. We're excited to be continuing to move this project forward. As was mentioned, in the coming months that we will be beginning work on modules 2 and 3 that have been described earlier today. So hopefully we'll take this data-driven approach as we continue to explore the potential use of ctDNA into the future. Also in the future, please be on the lookout for additional information for upcoming meetings that we'll be hosting, including our next data release on a different topic, although touched on in some of our sessions today around the use of real-world evidence. That will be on the 11th of September. It will be similar in the sense that we will be sharing new data from a real-world evidence pilot and have several leaders in the field that will be discussing the use of real-world response being obtained from electronic health data. I certainly also want to, one more time, thank all of our project partners for their efforts to move this partnership forward. We very much appreciate everyone joining today to present the new data coming out of these collaborative opportunities. You heard from a lot of our data partners on each of the different panels. We very much appreciate the time. You can see here on these slides that this is a very large collaborative effort with a large number of organizations and individuals that have shared their time and expertise across these different projects in order to develop them, analyze them, move them forward. Thank you very much. We would not be able to do this without this type of partnership, and we very much appreciate all of your continued contributions. So we look forward to seeing you at our future meetings. Thanks for joining us today. Thanks for joining us online. And until next time, I hope everyone has a great summer.